Well, first of all, thank you all for coming, the ones that are here physically and the ones that are here online. We have been organizing this event for a lot of months now, and I'm very happy that Olivier is here because he was one of the organizers. The other one, I think they are online, it's Suzanne Ditlevsen, Shigeru Shinomoto, Antonio Galvez, and Delphine Salort. And we are all here in the setup of IHP for a semester on mathematics and biology, which will last 4 months. It started at the beginning of January and will end in April. And we are very, very happy to open this conference about mathematical modeling and statistical analysis in neuroscience with Guilherme Ost, who is the invited guest of the whole semester for this biological and mathematical event. And so I'm very pleased to start with him, and he will give us a few words about his research and what he's going to do here for 6 months. And after that, we will have this nice video with your work of you and Claudia. Thank you.
Okay, do you hear me? Yeah, okay. So first of all, thank you very much, Patricia, for this very nice introduction. Thank you very much for all of you to be here. So as Patricia told you, I just want to say a few words before we start our real talk. As you might have noticed, Claudia didn't come because of COVID. And so to make the dynamics of our talk easier or better, we decided to make a short video. So just after my presentation, we will see the video and then we can discuss, okay? So first of all, I'd like to thank all the organizers of this very nice thematic trimester. So Luis Almeida, Vincent Cavez, Marie-Dominique, Benoit Partam, and Patricia Renaud-Bourret. And also the organizers of this very nice conference on mathematical neuroscience. So Suzanne Ditlevsen, Olivier, Antonio, Patricia, Shinomoto. And also I would like to thank the IHS and also IHP for providing me this very nice environment to do mathematics and try to understand some questions that I'm very interested in. So, okay. So what is the context of my research? So as you know, often if you look at the some project in neuroscience, you have a sentence like neuroscience is often referred to as data-rich, but theory-poor, right? So this means essentially that nowadays we have an excess of a massive amount of data, but still we observe many interesting phenomena on this data, but we usually they are poorly understood, right? So we need to do some theory or develop some statistical tools to make sense, to account for those observed phenomena. And this is essentially the content of my project. And what I want to do specifically is to try to answer 2 types of questions. The first one is what is written there to develop a new mathematical framework. We study the population coding for grid and place cells. So what does it mean? So in those 2 pictures, what you see here is the trajectory of a rat in a square environment in black, okay? And in the picture in both pictures, the red dots, they correspond to positions where a given neuron is more active, is spiking more, okay? And so in the picture on the right-hand side, you see that those positions are much more localized, and cells and neurons with this property are called place cells, okay? And the region is called place field. And on the picture on the left-hand side, you see that the structure of those prefed locations is much more richer, right? And in particular, they form a hexagonal grid, okay? And that's why they call those cells grid cells, okay? And one of the questions or the type of questions that we would like to answer here is, okay, first, we can think of as these 2 population of neurons as coordinate systems of the physical world in the brain of the animal, okay? And what we want to do here somehow is to say, okay, I take two positions, how close they can be, and in such a way that we still can distinguish between these two positions just by looking to one of these two populations, okay? And also, we would like to say if that the coding that we see here is optimal in which sense, okay? And so we want to formalize this in terms of hypothesis testing, which is very different from what people have been doing to study this type of cells. They have been doing using tools from information theory and all, and we want to say something different and try to compare with the results that we have so far, okay? And the second type of questions is, okay, so usually, so the picture here is just to remind us that most of the times, or always I would say, when we record the activity, the spiking activity of a given animal in a given experiment, it turns out that activity is always, the spike activity corresponds to a very small part of the brain, right? So in this sense, the information that we have is always partial, okay? And the question is that we want to address here, okay, when we have it, we look at this data, what can we actually learn from them, okay? Especially because the information is partial. And it turns out that even, I think it is just everybody is aware of this fact. When we go to the literature, you see that many of the statistical methods are not tailored for partially observed networks. So there are a few exceptions, and probably there are others that are missing here, so I apologize. I just want to say a few of them. So for instance, we have this paper from Mathieu Lehalle and Daniel Takahashi, where they have a Markov random field. They want to estimate the transition probability. They know that they observe just the activity to a given small region of the whole space. But here it's difficult to apply this in neuroscience because they speak about IID models, which is an assumption which is difficult to justify when we look at the data, right? There are also there is also a paper from Liederman, Adams, and Pilen where they have this Bayesian approach for a GLM model, which is also they want to model partial observed networks. But it turns out there is no theory there, okay? And then there is a paper from Aline, Antonio, Eva, and myself when we have a spike network of neurons. And we wanted to estimate the graph of interactions. We still have the results, which we have some results for a partial observed case. But it turns out the method, the algorithm is not computationally efficient, okay? And then more recently, there is a paper from Liu, where he studies Hox processes, which have been used a lot in neuroscience the last year, sorry. And he wanted to, so he has a random network, and he wants to estimate the density of connectivity in this graph, in this model. And even in the partial observed case, and it turns out, I mean, the downsides of this paper is that the intensity is linear, and if you know a bit about Hox processes, you realize that in this case you can only have excitatory connections, and so this is a bit not well suitable for for data, let's say, data analysis. And the last work that I would like to mention is a paper with Patricia and myself, where we have this large network of neurons, even an infinite network of neurons, and we wanted to estimate the transition probability, right? I mean, the probability of observing a spike given the past. We want to estimate this function. And we have some results to approximate this transition probability by using some given dictionary. The theory here is nice, but the problem is, as you might know, when we have these lasso results, we always rely on these restricted eigenvalue properties, which you need to check for each dictionary that you are using, and this may be difficult depending on the dictionary that you want. Okay, and I'm sure that so this is more or less related to statistical analysis of neuronal data, and I'm sure that we'll heard much more about this in the conference by Shinamoto, Eden, and Cass. Okay. So before just to conclude, I would like to mention that I've been working a bit also from, let's say, in the modeling of large and spatially extended networks of neurons. And one usual approach to do this is by means of mean field analysis. So you have a model, you give a description of how your neurons interact at the macroscopic level, and then you try to derive a macroscopic limit or equation to which will depend, of course, on how you impose the rules at the microscopic level. Okay, and so I have worked with Aline, Andres in a GL model. I'm sure that we heard about this model during the week. And more recently, I have worked with Julien, Eva, and Aline also when we have this a model for Hox processes with spatial, where the interactions taking into account also the spatial positions of the neurons. And we show that this process, so if I have a Hox process at a microscopic level, then at the macroscopic level, we ended up with what's called a neural field equation. And on Friday, I think it's Fridays, or at least the last 2 days are dedicated to objects like this, okay? And finally, I would like to mention that we also have studied the fluctuations of these spatially structured Hox processes around its limit, which is a neural field equation, and then we make sense of what's called a stochastic neural field equation. And we would like, in this context, we'd like to understand how we can use this result, as, for instance, to estimate the connections of your network. Okay. And then, as I said, we will heard much more about GL neural models during this week. So we'll have the talks by Fumier, Rush, Lechekba, Ipuzar, and also Morgan. And we'll heard much more about Hox processes and neural field equations in the Phi, Stanat, and phase talks. Okay, so now now I'd like to introduce our my joint talk with Claudia. Okay, so before we start, I just want to mention that this is a joint work with Noslen, Aline, Ricardo Freiman, Antonio Galvis, and of course Claudia Vargas. And I hope you enjoy. And after that, I will be happy to discuss together with Claudia. Okay. So.
I would like to thank Patricia Bourret and the organizers of the workshop on mathematical modeling and statistical analysis in neuroscience for inviting Guilherme Hort and myself to be here with you today at the Institut Henri Poincaré. I also wish to acknowledge the support of our funding agencies and that of FAPESP to the Center of Research, Innovation and Diffusion in Neuromathematics, Neuromat. The title of our talk is Retrieving the Structure of Probabilistic Sequences of Auditory Stimuli from EEG. This talk is part of the activities developed within the research project termed the Statistician Brain. According to this conjecture, Acting as a statistician, the brain makes predictions about future events in order to interact efficiently in the world. Predicting means anticipating outcomes. The idea that the brain makes predictions so as to anticipate outcomes is by no means new. It goes back to the pioneer work of Hermann von Helmholtz, a 19th century multi-scientist who introduced the notion of unconscious inference. Helmholtz conjectured that inferring about future events should be a crucial mechanism for learning and memory formation. Since then, it has been widely proposed that the brain assigns probabilistic models to samples of stimuli, also called the Helmholtz heritage in the contemporary neuroscience domain. Our goal here is to tackle the following challenge. Does the brain assign probabilistic models to sequences of stimuli so as to act in the world? Take, for instance, a sports person like Fabiana Müller, our Brazilian world champion in pole vault. As stated by Helmholtz, she improved her fantastic skills by training and adjusting her models of the reality. But can we transform the sequence of actions performed by this incredibly skilled person into a mathematically treatable object? And how to approach this question experimentally? A typical solution to this problem in neuroscience is to employ, for instance, simplified sequences of visual stimuli, such as the one depicting a human walk. As shown in this picture, it is possible to put a marker in selected points of the body and then extract only the relevant information that relates to the body movement. We then present to participants videos depicting these point slides while we record their brain signals employing an EEG machine. These EEG signals can be treated so that each recording electrode is considered as a node in a graph of interactions, depicted here as red points, and the vertices correspond to interactions between nodes. We then analyze statistically the graph metrics per node, such as distance, degree, etc., and we get very interesting results about how each node behaves in regard to its relatives during the point-like display visualization. Note that there is an ever-changing activity through time across the graphs per participant, as depicted here for 2 repetitions of the same video. However, this approach precludes to retrieve from the recorded brain signals a signature of the employed visual stimuli. In other words, there is no way to establish a formal relationship between the sequence of point-like displays and the graph of interactions. So, at this point, with the Neuromath team, we had some open questions. How to extract from brain signals the very structure of a sequence of events? How to establish a formal relationship between this sequence of events and the recorded signals? We started to think on a new experimental protocol in which we would provide to the brain a model containing information about the structure of a sequence of events while we recorded the EEG signals. We wanted a model that could be described formally so as to retrieve this model from the EEG signal. The chosen model was based on the work of Jorma Rissanen, who proposed in 1983 a class of stochastic models capable of compressing any sequence of symbols generated by a source. Rissanen introduced a new class of probabilistic models, namely the class of stochastic chains with memory of variable length, also called context-free models. The mathematical formalism and the experimental approach employed in this work is described in this recent publication and will soon be explored in detail by Guilherme. Shortly, volunteers were exposed to sequences of auditory stimuli where they had their EEG signals recorded. Stimuli consisted in strong, weak, and missing hand claps. We employed two different context trees that were called Ternary and Quaternary. In the Ternary sequence, a strong beat was followed by two weak beats, and in the Quaternary tree, besides, there was a constitutive silent unit separating the two weak beats. In both cases, eventually, the weak beat could be replaced by a silent unit, introducing a variability in the sequence of events. Following Rissanen's proposal, these sequences can be represented by context trees. Given the present symbol, the context tree represents all the possible past sequences with respect to that present unit. The transition probability associated to each context is used to choose the type of auditory stimulus appearing after that context in the sequence of stimuli. When we look at the EEG signal recorded during an experiment, there seems to be not much information across electrodes, as shown here in the right. In the neurophysiology domain, a trick is to repeat the same stimulus, say 1,000 times, and average the corresponding EEG response segments. By doing this, we get an event-related potential. The auditory event-related potential has certain regular temporal features allowing its identification. If a sequence of identical auditory stimuli is followed by a deviant sound or even by the auditory stimulus absence, there is a specific modulation in the EEG signal that is called mismatched negativity response. This marker of novelty, discovered in 1978 by Naatanen and colleagues, has been reported since then by several researchers in the field. Here, the deviant response is depicted in red, as opposed to the response to a standard auditory stimulus depicted in blue. 0 on the X-line corresponds to the stimulus onset. The red spot in the brain image in the right shows the location from which the mismatch response was recorded in this participant's brain, in this case, the left auditory cortex. As you will see, the mismatch negativity effect helped somehow to guide our analysis.
Hello everyone, my name is Guilherme Ost, and in the next few slides I will show you how we model the auditory stimuli we consider through context tree models and how we characterize context tree models. To this end, let me start with this simple example. So consider the following deterministic sequence 2, 1, 1, 2, 1, 1, etc., where the symbol 2 corresponds to a strong bit and the symbol 1 corresponds to a weak bit. Then replace each symbol 1 by a symbol 0, which from now on corresponds to a silent unit, and we do this independently with probability 0.2, producing, for instance, a random sequence that you see here on the screen. So in this case, it's 2, 1, 1, 2, 0, 1, 2, 1, 1, etc. This random sequence is an example of what we call in probability theory stochastic chain. And the stochastic chain we produce in this way is going to be denoted by X in boldface. And this is a shorthand notation for the whole sequence, which is X0, X1, X2, etc. And in this example, X0 is the symbol 2, X1 is the symbol 1, X2 is the symbol 1 as well, and so on. And by the structure of this auditory stimulus, we call it ternary. Now, let us observe that there is an alternative way of generating the ternary chain X. the chain X can be generated step by step, and this can be done as follows. Suppose we want to generate xn given the past xn minus 1, xn minus 2, etc. What we do is first we look at last symbol xn minus 1. If xn minus 1 is equal to 2, then xn by the structure of the chain X is going to be 1 with probability 0.8 or 0 with probability 0.2. Now, if xn is either 1 or 0, then we need to go back one more step in the past. So let's say that at time n minus 2, we have a 2, that is xn minus 2 is equal to 2. Then, by the way we constructed the chain X, we know that xn is going to be 1 with probability 0.8 or 0 with probability 0.2. Why? So remember, we want to simulate xn, so at time n we have a question mark. We want to simulate the symbol xn. And in this case, we know that we have observed at time n minus 1 either 1 or a 0. And at time n minus 2, we have observed the symbol 2. So by the first way we constructed the chain X, we know that at time n, we will observe a 1 if we didn't erase the deterministic 1, and this happens with probability 0.8, or we will observe a 0 if we replaced the deterministic 1 by a 0, and this happens with probability 0.2. The other possibilities for xn minus 2 is either the symbol 1 or the symbol 0, and by the structure of the chain X, we know that in this case, xn will be the 2, so that's why I have here xn equals 2 with probability 1 in this case. And we have a small drawing here on the right-hand side, just to explain this again. So we are at time n, we have a question mark here. We want to simulate xn, and at time n minus 1 have observed either 1 or 0, and the same thing for a time n minus 2. So we have observed either 1 or 0. And by the way we constructed the chain X, we know that at every 3 symbols we observe a symbol 2. So in this case, we must observe a symbol 2. The algorithm generating X is characterized by 2 elements. The first one is a partition of the set of all possible sequences of path units that we denoted tau. And this set tau is the set of the strings 0, 0, 1, 0, 2, 0, 0, 1, 1, 1, 2, 1, and 2. In the partition tau, for example, the string 0, 0 represents the set of all past symbols ending by the ordered pair 0, 0. And to give another example, the string 1, 1 represents the set of all past symbols ending by the ordered pair 1, 1. Following reasoning, the elements of tau will be called from now on contexts. Moreover, this set tau can be represented by a labeled and rooted tree, as shown in figure 1. As you can see, in this tree, the time goes downward. Each level of the tree represented a different time instance. Moreover, the leaves of this tree are the contexts of tau, and this tree representation gives rise to the name context tree. So that's why we call tau an example of a context tree. In the algorithm generating X, for each sequence of past symbols, the algorithm first identifies the corresponding context, W in tau. And once the context W is identified, then the algorithm chooses a new symbol A in the set 0, 1, 2, using a transition probability PA given W. In other terms, each context W in tau defines a probability measure on the set 0, 1, 2. Thus, the family of transition probabilities PAW with A in 0, 1, 2, W in tau
is the second element characterizing the algorithm generating X. The family of transition probabilities associated with tau are shown in the figure 2 on the right-hand side of the screen. And to give you an example, the transition probability indexed by the context 2 is given in the first row of the table appearing in the figure 2. As you can see, P0 given 2, that is the probability of observing a 0 after observing a 2, is going to be 0.2, and P1 given 2, that is the probability of observing 1 given that we had observed a 2, is 0.8, whereas P of 2 given 2 is going to be 0. That is, the probability of observing a 2 giving a 2 is going to be 0. Okay? So these are the two elements characterizing the algorithm generating the chain X. The ternary chain X is an example of what we call a context-free model. An important feature about context-free models is that the dependence from the past has not a fixed length. Instead, it changes at each step as a function of the past itself. This explains why this class of models are also known as stochastic chains with memory of variable length and variable length Markov chains. The class of context-free models have been introduced by Henri in 1983 as a universal system of data compression. And very briefly, a context-free model is a stochastic chain characterized by a pair tau p, where tau now is the context tree describing the dependence from the past of the model, and P is the associated family of transition probabilities indexed by the context tree tau of the model. If you want to know more about context tree models and their applications, here we have a few important references. The first is from Henri with applications to information theory, and then the second one is from Bierman and Weiner with applications to DNA data. And then the third is from Galvis et al. With applications to linguistics. And the last one is from Belloni in Buzzero, also with applications to linguistics. We also used another context-free model to generate auditory stimuli in our study called Quaternary. The Quaternary auditory stimuli is obtained by considering the deterministic sequence 2101, 2101, 2, etc. And then very similarly to what we have done to the ternary auditory stimulus, we replace in a IID way each symbol 1 by a symbol 0 with probability 0.2. By studying the algorithm used to generate step-by-step the quaternary auditory stimulus, one can check that the corresponding context tree is the one that you see here on the left on the screen, and the family and the associated family of transition probabilities are given in the table that you can see here on the right-hand side of the screen. Recall that in the experimental protocol Claudio described before, we have volunteers listening to a sequence of auditory stimuli while EEG signals are recorded from their scalp. We have seen so far how we model the sequence of stimuli as context-free models, and now I will explain how we model the EEG signals associated to a given sequence of stochastic stimuli. To this end, let Yn be the chunk of EEG signal recorded while a volunteer is exposed to the auditory stimulus Xn. For instance, in the image here on the screen, we have an EEG signal of a given electrode, and if we look at the green rectangle, we have that Xn is equal to 2 for a given suitable n. In this case, Yn is the chunk of EEG inside this green rectangle. The time domain of each EEG chunk is 450 milliseconds, from minus 50 to 400 milliseconds relative to the onset of the auditory stimulus. The model assumption is the following. The law of Yn depends only on the context W associated to the past xn, xn minus 1, etc. According to this assumption, for instance, whenever the context associated to the past xn, xn minus 1, etc. is 2, that is, xn is equal to 2, the law of the EEG chunk Yn is given by a suitable distribution indexed by the context 2. Now, if the context associated to xn, xn minus 1, etc. is, let's say, to 1, then Yn is chosen according to some distribution, which is going to be indexed by the context to 1, and so on. The EEG signal is then the stochastic process Y, obtained by concatenating the EEG chunks Y0, Y1, Y2, etc., constructed in the way we just described. From the statistical point of view, one of the main contributions of our work is to present a statistical procedure to estimate a context tree from a sample x0, y0, etc., xn, yn, where x0, x1, xn is a sample of auditory stimuli modeled by a context tree, and y0, etc., yn are the associated EEG chunks. And this can be done as follows. We fix first an integer k larger or equal than 1, and we do the following. 1. For each symbol A in 0, 1, 2 and string U of size k of symbols in 0, 1, 2, we define the subsample of all EEG chunks Ym such that xm minus k is equal to A, xm minus k plus 1 is equal to u1, etc., xm is equal to k. So to give an example, in purple here, we have examples of k equals 2, u is the string 1, 2, and A is the symbol 1. And the rectangles, and the green rectangles corresponds to examples of k equals 2, u equals 1, 2, and A equals 0. In the second step, we do the following. For any pair A and B, we test the null hypothesis that the law of the EEG chunks YAU and YBU collected at step 1, not 2, so this is a mistake, are equal. So here there are two possibilities. The first one is if the null hypothesis is not rejected for any pair of final symbols A and B, we then conclude that the occurrence of A and B before the string U do not affect the law of the EEG chunks. And in this case, we repeat the procedure with the shorter string U, which is U2, etc., UK. And the second possibility is if the null hypothesis is rejected for at least one pair of final symbols A and B, we conclude that the law of the EEG chunks depend on the entire string AU, and we stop the pruning procedure. In step 3, we keep pruning the sequence U1, UK until the null hypothesis is rejected for the first time. And in step 4, we call tau hat n the tree constituted by the strings which remained after the pruning procedure. An important step in the statistical procedure I just described consists in testing the equality of distributions for two samples of EEG chunks or segments. To this end, we used the projective method proposed by Cuestas Albertos et al. In 2006. In a few words, the projective method works as follows. First, we choose at random a direction, for example, a Brownian motion or a Brownian bridge, and project each function in the samples in this direction. And this produces two new samples of real numbers. And in the step 2, we test whether the samples of the projected real numbers have the same law or not. And we can use, for instance, here at this point, the Kolmogorov-Smirnov test. under suitable conditions, it has been shown in Cuestas Albertos et al. That for almost all directions, if the test does not reject the null hypothesis that the projected samples have the same law, then the original samples have the same law as well. The fact that the result holds for almost all directions allows us to repeat step 1 and 2 above for several directions. In this case, we decide not to reject the assumption that the two samples of EEG have the same distribution if the number of non-rejections of the projected samples over the different directions is sufficiently large. In our paper, we used in each test 5,000 directions, and we only rejected the null hypothesis that the distributions are the same if for more than 276 rejections are obtained over the different directions, and we refer to our second paper to the details justifying this choice. After applying the statistical procedure described above, per electrode and per volunteer, we obtained a tree denoted by tau hat n e v. So this is the context tree estimated in the electrode E of volunteer V. And by doing this, we ended up on each electrode E with a set of context trees that we denote tau En, as you can see here in step 1. We then summarize this set of trees in a unique tree called mode context tree, which is defined as follows. We compute the number of times each string W appears as a context in the context trees belonging to the set TEN. The context in the mode context tree, marked in red here in the image on the right-hand side, are the nodes that satisfy two properties. The first one is no node in the subtree induced by W has greater value than it. And 2, no node in the path from W to the root satisfies the property 1. Let me give you an example. In the image displayed on the bottom right of the screen, the string 00 belongs to the mode context tree because the nodes 00, 100, and 200, which are the nodes in the subtree induced by the string 00, they appear 4 times each, whereas the string 00 itself is estimated or appears as a context for 11 volunteers, as indicated in the number in red. And so this is property 1. Moreover, the nodes in the path from 00 to the root are estimated as a context for no more than 4 volunteers, as indicated in the picture. So the string 0 was estimated as a context for 4 context trees, and the root was not estimated as a context for none of the estimated context trees for that electrode. One can check that in this example, the nodes in red are indeed the contexts of the mode context tree, as we just did for the string 00. Very similarly, one can check that in this example, the nodes appearing in red are indeed the contexts of the mode context tree. And we can also see in this example that the mode context tree matches perfectly the context tree of the ternary chain, which was the true stimulus in this case.
So here I show the results for the quaternary context tree. This picture shows a sample of results obtained for a given electrode per participant for the quaternary context tree. We had 20 participants in this experiment. You can appreciate that there is some variability across participants. Similar results can be appreciated across participants for the ternary tree. Some variability can be identified across participants. A challenge of this work was how to average these trees across participants per electrode. As detailed by Guilherme, this was solved by estimating a mode context tree per electrode. This is a summary of our main results, published recently in scientific reports. Employing the mode approach, we were able to reconstruct a context tree per electrode across participants for the Quaternary tree in the left and the Ternary tree in the right, here represented as top views of the skull. The color code consists in a distance measure from the original context tree, where green corresponds to maximal similarity and pink maximal dissimilarity from the source context tree. As you can see, signals recorded from frontal and left temporal electrodes, labeled in green, were those who were closest to the original tree. These results are consistent with fMRI studies showing that the prefrontal cortex is involved in learning context-based statistics. So to conclude, we established the formal relationship between the structured sequences of random stimuli and the stochastic process associated to the brain processing of those stimuli. This was done by employing the new functional data statistical selection procedure just described by Guilherme. The obtained results support the conjecture that the brain effectively identifies the context tree characterizing the cells. Guilherme and I wish to acknowledge the colleagues that participated in this work. Thank you for your attention.
Thank you a lot, Claudia and Guilherme. I think there is time for questions now. Is there any question in the room? I have one. At the beginning of Claudia's talk, you were speaking about connectivity between the electrodes. And then your analysis for the moment is just analysis electrode by electrode. Do you have any idea how to take into account this connectivity between electrodes or brain areas inside the analysis?
Yeah, that's a great question. I don't know if I'm... okay. That's a great question. For the moment, I at least didn't think about how can we try to account for this, for what you mentioned. But maybe, I don't know, Claudia may have... may want to say a few words about that.
Yes, thank you very much, Patricia. Excellent question. We have been considering this possibility, but we didn't work yet on this direction. But I think it's an excellent suggestion. We can move forward on trying this approach. Thank you.
Especially the trees you showed at the end, they seem really different depending on the electrode. And if I understood correctly, the green ones are the trees that are the closest to the tree of the signal, right?
Mm-hmm. Right, exactly.
Exactly. Do you have any idea, for instance, why the ternary and the quaternary rhythm do not even involve exactly the same electrodes? What would be the cause of that?
Guilherme wants to discuss on that. Patricia, that's probably a mixture of the procedure that we have employed. But what is similar between them is that we do have the full trees reconstructed in the prefrontal electrodes, not all of them for the ternary, as you noticed. But the fact that we have a constitutive silent unit in the quaternary tree may have helped us in identifying more properly the original tree. So this is something we can discuss more.
Okay, thanks a lot. Another question.
Yeah, thank you very much for the presentation. Very interesting. Indeed, you have isolated a very interesting effect. My question was, how would you, do you intend to generalize to more complex types of stimuli, and how does it fly for more complex things?
Yes, very good question. Yes, we are now starting to build different types of context trees. For instance, the trees that we have employed have a regularity, regular base, a structured base. So it will be interesting to see what happens if you break down this regularity, this periodic, say, structure of the tree, and see how it goes for participants to learn this different structure. So we are now interested in going more on depth on the kind of stimuli that we have just built, Olivier. So this is our proposal for now.
Yeah, I just wanted to mention also that I know that there is, in Neuronal, there is an ongoing project where they have a similar protocol where they have a person playing a game. And then the game is essentially, Claudia can correct me, of course. I may say something wrong here. And the idea is that, so you are the goalkeeper, and there is you are the goalkeeper. So here, you are the goal, and then there is the kicker here. And the strategy of the kicker is given by a context tree. And then the person is playing. And then, of course, now the data is not anymore the EEG, but they are measuring the reaction time of the goalkeeper. And the idea, so again, you have either the left, right, or center. So that's the three possibilities. But the protocol is essentially the same, and so the idea is that try to by looking into the answers and the reaction time, if we could try to retrieve also the tree in the same spirit as we did here. So just to mention that there is already another ongoing project where they have different type of stimuli applied. Okay, so probably we should have a link here, but if you type goalkeeper game Neuromat, you'll find much more about this project. Okay.
Yes, excellent, Pierre. Thank you for bringing up this protocol that we are working also, and we wish to couple with EEG. This is the next step. Also, couple the goalkeeper game with the EEG, so that we can start looking at these prediction markers also associated to making a decision to go left, right, or center, and learning this sequence of events. Yes, thank you.
Other questions? Yes.
Hi, thank you for your talk. I'm neurobiologist, so I didn't understand all the mathematical part, but so I was wondering, when you were talking about this task where is motor task, okay? But so you were also mentioning about direction and decision making. Do you think that you will have a signature on the EEG, like a mismatch, like you were explaining, the one that you were explaining that is very, very clear to detect? But for the decision making, I don't think that you will have only one signature visible on EEG. And so what is your strategy in this case to solve this problem? Because it's all our problem in biology, so we have many cognitive part, and so it's not only just action and movement.
Yes, very interesting question. Thank you. So the idea would be to decompose, to choose the windows of over the EEG that corresponds to the moment the person chooses which will be the next step. And by choosing, he has to press a button. So we have this period just before the response selection. And then the game is structured so as he has a feedback of if he has given the good response or the bad response just afterwards. So we can just cut the EEG signals in periods and trying to find the specific signature that corresponds to the response, the prediction says associated to the response that the person gives, and then the feedback he has afterwards. I don't know if I made myself very clear, but if I answered your question, you want to comment on that?
I really understand, yes. That means sequence the different process step by step and see if you have a signature for each of them. Is it?
Yes, exactly.
Right.
Yes,
Thank
yes. Thank you.
Thank you. Other questions?
Okay, thank you very much for this nice talk. I have a question rather related to this decision-making problem and a little bit similar to the previous question, related to the previous question. You mentioned that in the other project, which covers studies more or less a similar approach, but applied to this goal-keeper problem, that the indicator that was considered was rather the reaction time. But the relation...
I'm sorry, go on. Sorry. Yeah.
Okay, but the relation between the input, let's say, in that problem and the output, in my understanding, is reflected rather on the decision, the final decision which is made, the performance result. Mm-hmm. So, and also maybe even this performance result is not sufficient, or only reaction time is not sufficient, and maybe other indicators are needed to be checked. And this was my question: how using only the reaction time, you think, shows or reflects the relation between the input and the output of the whole procedure of decision making? It's also quite high-level procedure process in comparison to.
Yeah, of course. I mean, this is of course. I will rely much more on Claudia's answer because I'm not really involved in the project. I just wanted to mention that there were another ongoing projects with close type of proposal that we were just describing. So I don't know, Claudia, if you want to answer the question.
Yes.
I don't know. I mean.
So we are looking at the performance of the participants. So the error rate, and this information gives us a measure of how learning evolves through time, of course, how the person is learning that context-free model, right? And we also are measuring the reaction time, which surprisingly gives us interesting information on how the person takes into consideration the errors that he made in the past. So this was just published as a doctorate thesis in our Neuromat, work done by Paulo Passos. And the next step now is to use information coming from the EEG while the person is doing the task, so that to try to disentangle these several, say, contributions of the moment the person makes the decision, makes the choice, and also how he reacts to the error that he made. So this is an ongoing project. Thank you.
Okay, thank you.
Okay, thank you. I think it's time we switch, but it was very interesting, and thank you both of you for doing the opening of this conference. Yes, we clap. That's the time. There wasn't. This is the 34 stuff were just us discussing about the sound level and stuff. So, as far as I understand, there wasn't. Oh, yes, sorry. There was one question on YouTube, maybe just one, who said, "Do you have any recommended textbook on the topic?" No, I think you just go to Neuromat's website and see every information that you can get. And so this is a very international conference because after Claudia, who was in Rio at 6 a.m., I think, now we have Shigeru Shinomoto from Kyoto who is speaking to us, and it's 7 p.m. for him, so thank you so much. And so we are so happy to welcome you for this second talk, and I leave you the floor, and then I come back for the questions. Okay?
Thank you. Thank you for Patricia for the introduction, and I'd like to start my presentation. And can you see my slide? Yes, it appears. Can you hear me, Patricia? No. Problem. Can you hear me? Can you hear me?
Yes.
Okay. Thank you.
Yeah, we can hear you, but not that much.
Okay. Oh, sorry. Okay, let me start my talk. And now it's okay. You hear me? Wait a minute.
I think you can try now.
Wait. Can you hear me?
Yes.
It's not good. It's not good enough. Can you hear me now? Okay.
Yes.
Okay, thank you. And let me start my talk. And thank you, Patricia, for introduction. And I had been expecting to visit Paris and looking forward to meet you directly. And but unfortunately, I was not allowed to visit you. And as a revenge, I and my wife went French restaurant last week in Kyoto. And let me start my talk and about the inferring monosynaptic connections from the cross-correlogram of the neuronal spike trains. And I'm Shigeru Shinomoto from Kyoto. And let me start with my, I'm sorry, my personal status update. And I just retired from the Kyoto University last March on the mandatory retirement age at 65, unfortunately. And but I was hired from by several institutes, including the ATR in Kyoto. And I am now working in the Brain Information Communication Research Laboratory. It's quite a nice institute. And I am enjoying the research and starting the new project. And I had been working making many toys. And it's sort of my hobby. And I made up a lot of toys and just uploaded several videos into YouTube. Especially expecting to introduce you Yeah, and so I hope I can do it in the end of my talk. Anyway, I had been working for the self-excitation issues recently, and let me start. Oh, sorry. No, sorry. Sorry, I will do it again. Okay, I have been working for the self-excitation issues, and particularly for the emergence of non-stationary fluctuation in the Hawkes process, and estimating the exogenous and endogenous origin. Exogenous are the external influences, and also endogenous self-excitation, internal self-excitation. And also we applied the Hawkes process to the analysis of the COVID-19, particularly estimating the time-dependent reproduction number from the series of the number of daily cases. And I had been working for, particularly working for the estimating monosynaptic connectivity quite a long time, more than 6 years, and finally made up a paper. And so that I'd like to introduce this in this talk. And contents of this talk consists of the 3 parts, and mainly I talk about the inferring monosynaptic connections. And if I have extra time, briefly, I just want to introduce a completely independent new issue, again, irregularity issue. And I will conclude the talk briefly. And the first inferring monosynaptic connections. And so knowing the neuronal circuitry in the brain is one of the ultimate goals in the neuroscience. And there were many attempts or endeavors for doing this. And particularly for the intracellular recording, around 2005, there are very nice, outstanding studies by Song et al. And Yoshimura et al. And particularly, I like the Song et al. Both. I like both. But anyway, for the Song et al. Paper, maybe you know, and they do intracellular simultaneous intracellular recording, and reveal the non-random features of neuronal connectivity. Because I like it, and I introduced this paper to my friend neuroscientist, but the response of the neuroscientist was the following. He laughed at, and the brain is non-random. It's of course. It's, I guess it's a typical response of the neuroscientist to this kind of a study. But it's very difficult to know the circuitry from using the intracellular, because the number of the neurons that can be recorded is quite limited. So but extracellular recording, number of recordable neurons are increasing exponentially in time. And there is a hope that we can have a circuitry of this kind. I guess this is my first, I first saw the circuitry of the real neuronal brain in this paper. And I was so impressed again and repeatedly looking at this figure. And pyramidal neuron, the triangle represents the pyramidal neuron, and the circle represents interneurons. And the squares are undetermined and difficult to identify. But anyway, I was so impressed. But by repeatedly looking at this figure, I feel odd because there are few connections between excitatory neurons. And so I believe the information processing, essence of the information processing is represented in the circuitry among the excitatory neuron. It's my personal belief, but there are few connections drawn in this figure. And anyway, I realize we know still little about the real neural networks. But anyway, from the extracellular recording, we can make an inference for the connectivity. It was more than 50 years ago that we can, the Parker, Gerstein, and Moore suggested that we can make an inference for the connectivity using the cross-correlation diagram. And if the cross-correlogram exhibits a peak, it implies that there might be excitatory connectivity, and dip implies inhibitory connectivity. Meanwhile, I met George Gerstein in Kyoto in 2007 and made friends and visited his home and chatted and learned a lot. He's a very nice, impressive person. And as I worked in the extension of their work, I wanted to report my result, but unfortunately, he passed away a few years ago. But then, anyway, their original method is not much efficient for the analyzing the real spike trains in vivo, because there are large fluctuations in the cross-correlogram. And even if we take into account the possible statistical fluctuation, it was the original method tend to make a false positive connectivity inference. And in order to avoid this kind of influence, the Toyama et al. Suggested the shuffling method, and Amarasingam et al. Suggested the jittering method. And the previous the circuitry was drawn by this jittering method by Amarasingam. He was a co-author of the that paper, Fujisawa's paper. Anyway, we applied the generalized linear model to the cross-correlogram and adapted the function AT, smooth function AT, to the cross-correlogram to exclude external influence and try to detect a genuine connectivity between the two neurons, J12 and J21. And we were able to avoid making the false positives and so on. And in order to test suitability of our method, we made a simulation of the large network of the 1,000 Hodgkin-Huxley neurons or 10,000 Leaky integrated fire neurons. And we applied the cross-correlation method. And as I noticed, it made a lot of false positive connections. And Amarasingam, their jittering method was very cautious and carefully avoiding the false positive, but instead made a false negative inference. We tried to balance between false positive and false negative and made a reasonable estimation. And we also made a theoretical consideration about how to say recording duration we need for estimating the connectivity, because using the small number of spikes, it is very difficult to make an inference for the connectivity. So we need a large number of spikes. And so the necessary duration critically depends, strongly depends on the firing rates of the presynaptic and postsynaptic neurons, and also postsynaptic potentials. And for instance, for making the inference of the excitatory postsynaptic potential of 1 millivolt, we need a fairly large, long recording time. And then we applied our method to rat hippocampal data, and using the partial data, 10 minutes, 30 minutes, and 90 minutes. And we observed gradually by increasing the recording time or using longer spike train, we were able to increase the number of connections inferred. And also we applied the simultaneously recorded data between CA1 and entorhinal cortex. And this is a connection matrix, and also made up a connection diagram. This is a comparison. We have no ground truth. And in addition to that, recently, we also constructed the convolutional neural network model for estimating synaptic connectivity, and applied it to the rat and the monkey data, and obtained a large network. And I want to start analyzing the networks obtained. And I wish to introduce. So we made up a application program and uploaded in my website. And for instance, it's impressively for the 15 neurons. And the computation is impressively fast, and obtain the connectivity matrix. And we can obtain a lot of the cross-correlograms simultaneously. And I hope you could visit my website later. And by the way, Patricia, are you hearing? Can you hear me? Anyway. I hope you are hearing. Okay. Are you? Okay.
Do you hear me when I speak?
Yes, and yeah.
Okay.
Can you hear my talk?
Yes, that was fantastic.
Oh, okay. Thank you very much. Yeah, just I am anxious. Okay, I'm going. And just introduce independent new issue just briefly, and the spiking irregularity. And let me start with my old story. I introduced metric of local variation and tried to analyze data, and they estimate the firing irregularity more aptly than the classical coefficients of variation. And if you see the Louis Vuitton, please remember mine. And anyway, we applied these metrics to the neuronal spiking in the monkeys. And it had been, as it had been suggested, the neuron is a person spike generator, and the CV is distributed unimodally around the unity. But if we apply the local variation, it has a bimodal distribution. And if we look into the individual spike frames, it is one group is actually irregular, but another group is rather regular, we realize. And we collected many data from the various cortical areas, and realized that firing irregularity is different between the different functional area. Motor, premotor tend to be rather regular, and visual and prefrontal tend to be rather random or bursty. And we also collected more data from the cat, rat, and mouse, and including the French researchers, and also French, yes, French researchers. And anyway, so as you know, the Corbinian-Brodmann classified cortical areas into the different areas according to the cytoarchitecture, and it turned out that his classification is strongly correlated with the function of each cortical area. And what I did was it was strongly firing irregularity is strongly again related to the correlated with the function. And because the structure is different, it is natural that we have a different signal. New issue, just briefly, and I started to analyze cardiac signals and cardiac disorders, and you may know. And I collect, it was difficult to collect data, but we finally obtained the thousand pulse data from the ECG data, and plotted onto the plane of the log LV and the log CV. And you know, atrial fibrillation and premature ventricular contraction and premature atrial contraction. And atrial fibrillation, it appears atrial fibrillation is rather serious than the PVC and the PAC. And we realize that is the placed, and atrial fibrillation is distributing rather differently from these diseases. And I am a metric geek, and I again invented the new metric that I call the local global variation ratio, LZ, and found out they can be clearly classified in terms of this local global variation ratio. And particularly the value 3/2 it has some mathematical meaning and particularly interesting meaning. Anyway, if you are interested in, please have a look into my preprint. And finally, I wish to conclude my talk. And most important thing is that I am still alive, and I am still enjoy studying. And the newer thing is I started YouTube, and let me introduce the one just one final introduction. Oh, this is my YouTube video, and for instance, sorry, this is. I made up a lot of the funny toys after the retirement, and every non-working days I am making many toys. I am happy if you visit my my YouTube video, and thank you very much. Anyway, return to the previous. Anyway, thank you hearing my talk, and that's it. That's all, and thank you very much for your patience.
Well, first of all, let me just say that I think all hope that you will still be alive in many, many years, and you will have much more toys to do, and put electrodes on rats that are watching your toys and understand what their spike trains are saying when they are watching your toys. I'm sure it will be great. Is there any comments or questions in the room? Yes. The biologist is speaking.
Thank you very much for your talk.
Thank you.
So we are glad that you are still alive, as Patricia said. I was wondering, so when you were explaining at the beginning that with patch you're doing cross-correlograms, so it is easy in this case to know that to know if the two neurons are connected, because you can activate or inhibit directly.
Yes.
As soon as we are in extracellular, that is much complicated. And I was wondering, when you did your connectivity between the entorhinal cortex and the C1 region, is it direct projection that you were studying, or are you infer? Because it's a discussion that we have very often with Patricia. Are they physically connected or not? Are you inferring a connectivity? And so how do you do when you're doing this?
Thank you very much for your interest. And let me come back. Unfortunately, this data has no ground truth, and but at least we can say that the neuroscientists classified, tried to categorize them into the this excitatory or inhibitory neurons according to the based on the waveform of the pulses. Thin spikes, the inhibitory, and so on. And so that they have a at least they know they have a guess about the excitatory or inhibitory. So roughly speaking, they are in accordance, and we cannot say if they are really the monosynaptic, but by having a look at by looking into the individual cross-correlogram, we feel that like they are like a monosynaptic. It's a few millisecond delay. So this is just I personally believe it is real monosynaptic connections. But there is no proof. But English, the neurophysiologist English, his name is English, and they have some more, how to say, reliable data that contains the information for the real connectivity. So it might be interesting to check if our method is consistent with this kind of ground truth.
Okay, thank you. So you're taking in account the spike, the time of the spike train actually, just to be sure that they are connected for the moment. Yeah.
Yes, and also excitatory and inhibitory difference. And again, my LV, concerning LV, these are the how to say, putative excitatory and the putative inhibitory neurons have different, entirely different irregularity, my LV. And that is also supportive for our result. Yes, thank you very much.
Other questions in the room? I have one which is a bit linked but more mathematical to what Ingrid said. Could you go back on your formula where you are estimating J12 and G21? I don't remember it. Yeah, this one. This is this one. I was wondering, f of t, is it a function that you have, and a of t is the same? Are they functions that you have to estimate or something that you take as known because of some non-result? And in particular, that's why it's linked to Ingrid's guess. This formula somehow says that J1 and, well, neuron 1 and neuron 2 are alone in a world without connection to outside world, whereas in fact they are embedded in a huge network. And I think that if they are not corrected directly, it means that the other connection have an influence which is much smaller than that. And I was wondering where it is in the formula. You see what I mean?
It's difficult to understand. F of t, for instance, the shape of this kernel, the connection, the influence, I put it some given function, and for f of t has some latency, given latency of the arrival of the influence, and also it has a few millisecond influences and so on. And so that I put the exponential function as f of t. And so this is just given by hand. And just we try to estimate that this the real number, the strength of the connectivity, and also its statistical significance for the presence of the connectivity we discussed. And is this what does this?
Yes, just so that I'm sure to understand, does it mean somehow that what you call the filter FT, you tuned it to be of a few milliseconds so that you cannot see connections that are not monosynaptic? Is it where you kind of discard the ones that are not there? Okay, that are more complex.
Yeah.
Okay, fantastic. Thank you.
Thank you.
Other questions or comments? I just need to check also internet, but if there are any. Let me just check internet to be sure that I have no more questions. And
Yes, may I ask a question?
Yes.
Of course.
So thank you very much for this beautiful talk and very interesting data. My question is also biological. I was wondering what would be your interpretation of these differences in cross-correlation regularity across brain areas. How do you interpret these results? Thank you.
Cross-correlation across the brain area.
Yes.
You are asking. Yeah, I am very curious. Previously, I did the spiking irregularity analysis for the across the brain area. Actually, I am more interested in the different connectivities. How to say the style? I guess the connectivity. Structure is different between the cortical area. So this is ultimate the purpose. And so this our method is just a preparation for doing the analysis. And I started to collect the data recorded from various cortical area. And if some of the audience has interesting data, please let me know, and we can collaborate. Thank you very much.
Thank you.
Okay, I have a question from YouTube. I'm not sure I understand it, so I'm trying. I hope you will understand. This person is mentioning first a paper by Ushiba in 2002 about peri-stimulus time histogram using Monte Carlo. And with respect to this work, is asking if there is something fruitful to investigate around random walks model with respect to GLM and cross-correlation histogram. So I don't know if you know this paper or if this question makes sense for you. And the people online, if you want to precise, I would be happy to.
Yeah, I'm sorry, I don't know
Okay.
no, Ushiba maybe Japanese, but I don't know. Sorry.
Okay. I don't see more question. Okay, is there any other comments in the room? Okay, so I think first we thank you a lot, Shigeru. And so the conference will stop now, and we come back at 2:30 for the talk by Ingrid Betus and Alexandre Musi. And they will be physically here. Okay, goodbye.
Bye.
Elle est positive?
Alors elle est pas? Elle l'est pas? Elle a pas son résultat?
Okay. So, thank you all for coming back for the conference. We will start a program that will happen every day. Like each afternoon will be dedicated to a certain mathematical tool. And on the morning after that, you will have talks by younger people. And also there will be a discussion where we will be very happy to all discuss about the tool at hand. And so this afternoon is dedicated to statistics and machine learning. And for each of the 4 topics that we choose, so there is this one, tomorrow it will be algebra and geometry, after that it will be probability, and the last afternoon it will be on dynamical system and PDE. For each of those, we choose a first talk where it's a very nice collaboration between a modeling guy, say, or girl, and a biologist, so that they can explain how they interact on the specific topic with the tools of the afternoon. So I'm very happy to start with 2 people from Neuromod in Nice, Alexandre Musy, who is a computer scientist, and Ingrid Betus, who is a neuroscientist, a cognitive neuroscientist. And now it's your turn.
Thank you, Patricia.
Okay, thank you. So can you hear me? It's okay. So I will present you this collaboration with Ingrid. And the collaboration is about, many people are working on this topic, but maybe not at the learning level, but trying to link the behavior to the neuronal code. But here, the idea is to look at the behavioral learning through different individuals, and mainly it will be rats, as we will see, and to link it to the neuronal code. And to do that, we will use cognitive models or different cognitive model candidates, and we will, at a computational level, select some or say that it is not close to what maybe the rat is thinking. And we have different granularities. By granularities, we mean different pieces of trajectories, as we will see in the presentation. So this is the plan of the presentation. Ingrid will present a first introduction to animal learning experiments in the context of learning. Myself, I will present the computational modeling of the problem we identify. And finally, we will show the results and have a discussion on these results. So now.
Okay, so first, I will introduce to animal experiment in cognitive neuroscience. So as you understand, I'm the biologist who don't understand the math at all. No, now I'm trying to understand. So you already heard this morning about EEG and also single unit activity, but just to briefly explain that we are recording in our lab is different type of signal. So as you have seen this morning on the first presentation, you can record on the scalp of the animal that we label the EEG. So it's exactly the same electroencephalogram that we have in human. And so if you implant your electrode a little bit deeper, so that means inside the brain, you have also these oscillations, which are the rhythm, and its name is local field potential. Because as Claudia said this morning, so on the cortical surface, so at the cortical level, we have pyramidal cells which are very well distributed. So I remember that this morning I said in biology is not random. It's not random at all. The neurons are aligned, and it's because the neurons are aligned on the cortex that you have this oscillation, because it generates an electric field, and so every time that is going on the heel and on the deep all, you have this oscillation. So actually it's the recording of a population of neurons. And so Alexandre asks me all the time, how many neurons can you record? So it depends on your electrode.
And the second type of signal that we can have is the single unit activity. So the second talk that you heard this morning on the cross correlogram, it was especially on this type of analysis. So usually in neuroscience, we can implant that we label a tetrad. So it's quite useful, the tetrad, because with the triangulation of the signal, we can isolate the single unit activity, which does mean the spike of the neuron, the action potential of the neuron, which is completely different of the LFP, which is the sum of activity around the neuron, not only the spike, also that you have on the synapse. It does mean the EPSP and the IPSP for excitatory or inhibitory postsynaptic signal. So the signal between the both are completely different. So the LFP is the sum of all these signals, and the single unit activity is the signature of each neuron. So yeah, this morning, for example, Guillermo was talking about play cell, so it's the activity of specific neuron. And with this tetrad, we can have the local field potential. It depends on the filter that you put on your electrode. And so Buzake did a nice review where he said that in the cortical region, you probably register until 1,000 neurons on the LFP. And if you are recording the single unit activity, it does mean the spike of the neuron. So this is actually that we are doing in our lab. And the second thing is when you are doing these in vivo, because it's completely different that you have seen this morning with the second talk where they were talking about patch clumping. So it's on the single cell, you extract the brain, and so you can record in a Petrish plate. So when you're doing in vivo, that we want to do is try to isolate the spike correlated to the behavior. And which type of behavior? So here, for example, with the tetrad is easily to understand. You have 4 electrodes bent together, twisted together, and it depends, it's like a microphone. It depends if you are close or far from your neuron. And because they are twisted together, you can recognize and isolate that we are labeling the spike sorting. And the spike sorting actually will tell you if you have 1 or 2 neurons. Because of course with a microphone you can record a neuron if you are close, but if I'm here, I'm still recording the same neuron, but I'm far away. So that's why you have to discriminate between one and another one. So is that we are doing in the spike sorting. And so you are better than me. So for example, you can isolate with the peak of the spike. You can isolate with the amplitude of the spike, or with principal component analysis, you can isolate the different neuron. Okay. In biology, when we have that, after we try to correlate to an event, to a stimulus, to a behavior. And that is very interesting, is to try to interpret what does mean, or does this spike will be related or connected to a stimulus. For example, here you can see is very simple association, is that we label a Pavlovian conditioning. So for example, if you give a cheese to a mice, actually we're not given cheese but a chow, so if you give a cheese, you will have some neuron that you can see here on the raster plot. So this is the trial here, and if you are doing the summation of the trial, you have this graph here. And this activity, it does mean that, okay, when the cheese appears, the animal has a reward activity. We can label a reward activity. And very classical things which has been found by Schultz, it was the processing activity of a dopaminergic cell which predict that the reward will arrive. And so, for example, here you can see that at the beginning, when you place the cheese in front of the mouse, the firing rate of the dopaminergic cell related to the reward will activate when it sees the cheese. if every time that you put a cheese, you have a bell, it's like a condition, okay, and a conditioning. And so after all, when you will just ring the bell, the activity will precede the cheese. So it does mean that you have an association. But for this type of association, it's very short. You need just 1 or 2 associations. So it's simply, but when you are talking about learning, you need to increase the level of complexity. Second level of complexity is the navigational learning. What does it mean? It does mean that you are not constraining the animal in the Skinner box or in a box. So for example, here I will explain the task. Is very simple. You have a T-maze, and so the animal during the sample, he has no choice. He turns to the left to access to a reward on the top of this maze. And after a certain delay, you can give him a choice. And actually here, the rule is you have to shift. So it does mean win shift task. It does mean that if the reward was on the left, it does mean that during the choice, it will be on the right. So I will just show you a little video. As you can see, the mouse going super fast. So here, the mouse during the sample went on the right. We closed the door to force the mice to stay. The mouse hit already the reward. We're taking back the mouse, put it back on the start box, and you will see as soon as you open the door, and if you give the choice between the 2 arms, ta-da! The mouse knows already where it has to turn to find the reward. Okay, so what are we doing in terms of analysis with this type of behavior? It's a learning process, it's a long process, so we have the term of learning because we are interested in the learning. So it's a long process, it's taking age to arrive to this. Don't think that in one day it's enough, no. It's a really long, but so we have to define like flag, like even flag. And usually that we are doing is we just put a choice, a point here, and it's a flag. And for example, we will try to correlate the activity of the neuron when the animal passes the choice point to see if this activity will predict, for example, if it's going to the right or if it's going to the left. You see? But here, it does mean that it's still constrained, okay? Because the animal, you have to leave in the box, is going to do the sample, you're taking back, and after you give the choice and you're taking back. You don't leave the animals going around and find by itself and understand by itself. So, in our experiment, we would like to question the learning, and the second thing is ecologic learning. What does it mean? Sorry, I forget to talk about this. So, for this type of experiment, sorry, I go back to this experiment. So, usually that we are looking at is Peri-Event Histograms. What does mean a Peri-Event histogram? This morning you heard about cross-correlogram. Cross-correlogram is activity of a neuron compared to another neuron, right? Here, you are looking at the activity of a neuron compared to the choice point. You see the red line will be the choice point. And here we can see that, for example, we have a neuron which is firing just before the animals arrive on the choice point. Okay? And we can have a summation of these average rates, spike per bin, and see, for example, if we have an activity related to the correct choice or to the incorrect choice. It's very basic analysis, and it's usually that we are doing in the first analysis. Okay, but as I told you, now we want to increase the complexity. We want to leave the animal learn by itself and discover by itself where is the food, what is the role of the task. So for that, we imagined a new protocol, and it was, we guess, more ecologic and closer to human learning, with no even flag to correlate to neuronal activity. So it does mean that you will see what is the task. It does mean that we can't dissociate the type of cognitive process that are doing the animals. We can't dissociate, okay, now is the time that he's deciding. Now is the time that he remembers where he has been last time. Now is the time that he thinks that he has to turn to the right because he went to the left previously. So all the cognitive process are mixed, and we don't know exactly where on the maze where the different cognitive process. And at this stage, I was stuck. So that's why I call Alex and say, okay, I need you. I can't analyze the data because at this point, with the tools that we have in biology, it was not enough to explain and to try to understand the different behavior. So briefly, I will show you the task that we're using. So the task is an alternative task on a T-maze. So as I told you, the animal is not constrained. We place the animal at the bottom of the T-maze, and you will see he has to go whenever he wants for the first time, to the right or to the left. At this point, he will receive a sucrose pellet. He has to go back, return, go back to the central stem, and if he went to the right, he has to go to the left. So that's why we label alternative task, because he has to go to the right, remember that he has been to the right, go back on the central stem, and go to the left. So it's very useful, this type of task when you want to study learning, because we don't interrupt the animal. The animal is doing that he wants. So that means at the beginning we have a lot of errors. He has to try to understand by trial and errors, so it's very nice, but we have a lot of information, like decision making, like memory, and different things that we want to compute and we want to dissociate. And thanks to Alex, now it's possible, so you see the transition is very good. So during the same task, that we did also it was recording the spike activity. I told you just before, we have two types of signals. We have the LFP and we have the single unit activity. So during this task that we did, we were recording the single unit activity of the neuron, and we did that in collaboration with the LNC, which is the Laboratoire des Neurosciences Cognitives in Marseille, with Francesca Sargolini. And so you can see our respective students here, Maxime Villet and Julien Poitreau. And so they were training the animals, but they were freely moving. And so we were recording the spike, and after spike sorting, we call Alex, and he will tell you that we did.
Yeah, okay. It's not thanks to Alex, it's a team, okay. Ingrid said a lot about what were the problems, but so as the animal is free to choose the series of actions, so there is both action decision making and error origins which are unclear. So if you look at here, it is a percentage of success of the task. So 2 successive correct paths achieved for one trial. And here you have the session numbers. And you can see that, okay, there is a learning curve at the beginning, but even the whole time, the behavior seems really erratic if you look at it. And even if we try to separate during the learning phase and after the learning phase, defining, for example, this point as being, so we were searching for different definitions, but maybe 80 or 90% of success over 30 trials. So at this point, we know that the animal knows exactly what is a task. But despite this fact, you can see that it will continue to change the behavior and to do not want to get always the food. So there are many interpretations. Maybe the more plausible for our colleagues is that there is an exploratory behavior of the animal. He wants to explore, but we don't know. And so from this behavior, it is quite difficult, and even discussing with the biologist, to interpret the decision-making process. So as Ingrid said, even when we were discussing together, it was not clear. So is it, for example, you have different representations in the environment that the rat is perceiving as important for solving the task? So is it that, for example, the last pellet he obtained? Is it the next one? Is it the next action, remembering the last turn, or the next pass? And the order, the sequence of choosing these different representations or cognitive processes, the dynamics, it was very unclear, and it was a problem. So again, so the cognitive process was unclear, and also it was not possible to link the behavior or to make more explicit this behavior thanks to the neural activity, because we didn't know exactly what was important for the rat and how the behavior is timely bounded. When, for example, for the rat an action is beginning or ending was really not clear. And so, so now the idea was, okay, let's say we have the behavior of the rat, we can observe, we have different measures of this behavior. And now what we want to achieve is, thanks to the behavior measures, the activity, the different activities of these behaviors, we would like to infer the different cognitive processes and the different links between the processes. And we would like to test different models together. And once we identify the different cognitive processes, we thought, and we'll see that we were not so wrong, that it would be possible. I don't know if the questions are now or after, whatever. Yep. Okay. Yep.
I guess I think I might have missed what the constraints on the task actually are. So, you know, what's the setup? What are the rules that the animal has to learn in this setup, and you know why the particular design?
I think I will explain more in the next slides.
Great, thank you.
You tell me at the end if it's not the case, if I'm not clear about that. Okay, thanks. So. So yeah, the idea was, okay, so if we are able now to identify the different cognitive processes, the actions that the animal thinks are important, and for example, the different representations of the environment, the different states of the environment perceived by the animal, then we will be able to try to analyze the neural activity. So this is a four-step method where we define each model has a different cognitive representation, so we have different models. We run these models, we test them through the learning process. After we apply model selection, we try to find what we think is the best model. And finally, we perform the neuronal code analysis. So to do that, it is a new method, I will say computationally, that I'm using, compared to usual, for those that know about it, usual reinforcement learning, where usually you have no duration, really, of the actions that are engaged. And the goal of the algorithm is to find one best strategy. And here instead, with Arca, we would like to get not only actions, but maybe some components that are more general. It can be neurons, it can be actions. So at this level, it is still action. But we would like to compare the different strategies selecting model components. So we split the behaviors, and as you will see, in different components. And we'd like to see how the animals select these components, these actions, defining the whole behavior. And another dimension that we added is the time, the temporal activity, as I will show, and which is measured as a duration, a contribution of local components, the different choices of the animal, with respect to the behavior. And so now, these components, as I said, we were taking action. The notion of granularity will be important, and I will describe more precisely what is this. And it is a notion of segmenting the different trajectories of the animal. It can be a long pass, or it can be a single turn. And so the duration was important. So it was really a question beginning the project that can we implement and compare these different cognitive models using this activity-based credit assignment. So this is, as I said, a collaboration. And Ashwin is a PhD student working on this project. And it is still, Ingrid is co-supervising, and it's a collaboration, as you will see, with Patricia and Francesca Sagolini from Marseille, which is a neuroscientist. So here, for example, you get the probability of choosing the different paths, of choosing one path that Ingrid showed. You remember, so it was going from one pellet to the other pellet, alternating. And there was a probability of choosing this path. And here you have the different trials. And in dashed line, you can recognize it is the same rat, that you have your erratic behavior. And we started with an usual reinforcement learning algorithm, which was converging like if the rat was a rational agent, always taking the best choice. So you can see that it is not really erratic in the behavior. And more we increased with different parameters the different Arca models to finally, in red, follow better that curve. Okay, so let me go back to the experiment. So Ingrid showed the experiment. So the behavioral task to learn is this. The rat needs to learn to alternate these two paths in blue. So it needs to, for example, come here, take this blue path. It will obtain the food if the path is good, if the trajectory is good. And after, to go back by the red trajectory, and it will obtain the second reward, the second pellet. And so basically the task is these two alternating trajectories. It is, as we will say, what we define as an episode is corresponding to the cognitive tasks, the behavioral tasks that the animal needs to learn. So what it seems to be important for the animal are the rewards, positions, and the decision choices. So basically, we identified two different choice points. And so if we rephrase it, the rat needs to infer the association between the trajectory and the rewards to earn more rewards, more pellets to eat in the maze. So again, so this is a schematic and graphical language we define for representing the problem. So you can think of your T-maze with dashed line. And on the left, you will have the feeders providing the pellets of the food. And you recognize here your two alternating paths. So one first problem we had is, okay, so let's say that at the bottom intersection, the rat takes the green action, so wants to turn right. So depending on where the rat is coming, it should be that this action is represented differently. So it is where we added the notion of the state of the animal. And one thing that seems to be important is that the state is described by the head direction. So the rat, for example, you will see in the different graphs that the rat is coming at this intersection. And the state is described also by the rats. The rats remember the last feeder they are coming from. Okay, and so after you have the action that brings the rat to another state. The notion of granularity I was introducing before was we have, for the same trajectory, it was not clear which actions are cognitively important for the rat. So you can see that we splitted the actions in different components also. So more than the notion of states, we have for actions the different segmentations, granularities of the trajectories. And so what we designed using this graphical language is represented here, where here on the left is a model which is called the Ternary model, which is a finer granular model described. And on the right, the higher granular model, which is called the path model. So let's start with the path model. You recognize in red and blue the correct path. And so the rat needs to understand that it needs to go and to alternate between one feeder on the left and one feeder on the right, taking this long path. And these correct paths are described also here in red and blue. And this one is representing the turn model. So at the turn model, you have elementary actions. So for example, you start here from the left feeder, you decide to engage an action to go on the right. So you stop at the first intersection, you represent that the rat is here, and after you take another action, and so on. And in between these two representations, these two granular models, we have what we call hybrid models, which are representing both paths, I would say, and turns inside. And so, for example, we will come back to these two models, which are hybrid 2 and hybrid 3 models. You can see here. So they are basically the same for the correct path. You can see here that the rat is stopping at the same intersections, but they differ for the incorrect actions that the rat is taking on the left here. And also we have two other models, so it is a total of 6 models we were able to test. And here, the same way, the correct paths are basically the same, but for the incorrect choices, it is more the central arm choice that is in one case, and in the other case, it is the top intersection choice that is chosen. Okay, so now that we have all these different models, was the definition of the Arca model, which was already defined but not adapted to this maze problem. So it is how we define it. So we define the session number. So each session is a particular day that Ingrid was performing the experiment with the rat. And inside, you have the different episodes. So remember that an episode is the basic behavioral task the rat needs to learn. So alternating two times a pass. And finally, you have a trial number. So each time that the rat is trying a different action from a state. So the state is depending on the episode, the session, and the trial. And finally, we have a probability of taking the different actions as a softmax function, which depends of what we call the credits of the state actions of the animal. And the state actions of the animal is defined here. And so it is basically an accumulation of the different credits plus a learning parameter alpha, where we have the sum over the different trials of, okay, am I taking these actions and these states? So it is a state action that, okay, over many trials, I will increase finally this credit. And it is depending of the reward. The reward is, during an episode, do I receive 0, 1, or 2 pellets? And the activity defined here, I was speaking in the introduction about duration, is basically the duration of one trial over the duration of the whole episode. So this is for increasing the credit of the different state actions. But in between, we have also a decrease in between the sessions, kind of forgetting of the rat. So we called it a discount because maybe it's not really forgetting, but it's representing something moving in the decision process of the rat that we noticed also in the data. And so between sessions, there is a decrease for all the different state actions. Here, there is an increase of only the selected state actions. And here, for all the state actions, their credit is decreasing. So let's have a look now to the results we obtained. So we were comparing all these models. So the pass I was introducing, the turn, the different hybrid models. Here we have the different rats. So just the last rat has an unusual name because we were bored with these numbers. And so finally, you can see that looking at the negative log likelihood and comparing these different models, most of the rats have hybrid 3 model. So we let the numbers tell us and the simulations. And only one seems to have the hybrid 2 model. So I will come back to this difference after, but next you remember that our goal was, okay, so we had the behavioral level data. We are now able to have some kind of good cognitive models. So now is the time that, okay, we identified hybrid 2 and hybrid 3 as good models. Does it tell something at the neuronal model? And here I was discussing with Patricia, and I said, okay, what do you think about the computational model? The simulations are telling something, and maybe it can be rephrased by that question. So you remember that I told you that hybrid 2 and hybrid 3 intersect correct paths at the bottom. So they have the same bottom intersection. So I was asking, okay, does it mean that the rat coming at this bottom intersection, not on the other one and from another direction, so and coming here or here, does it mean that it predicts to go to one feeder at the bottom or just before the intersection? So it means that when it was my assumption, when it is in the central arm, it seems to be thinking about, okay, I know exactly where I'm going. And before, I don't know. So Patricia developed a test for that. And to look at the neuronal code. The neuronal code consists in the difference of firing rate. And so the test was, is neuron firing rate different between the red here and the blue path? So the blue paths here are the two correct paths. And here the red one is a feedback bad or incorrect path. And so we tested, or more precisely, Patricia tested two different things. One is, okay, is it the case in the left and right arms? So is it that when the rat is in the left or in the right arm predicting what it will go? Or is it when the rat is in the central arm that it predicts by the neuronal code where it is going? And so finally, we were very happy with that. At the beginning, she didn't find that and thought the hypothesis was wrong, but it's okay, it's usual. And so you can see that in the left and right arms, only 3% of neurons are coding. In the central arm, 22% of coding neurons code for this behavior. So it means that the firing rate increases or changes, it might be better, that increasing, not only increasing but also decreasing significantly. And another interesting result, we were in front of the computers in real time and said, okay, look at where, at which phase is it? Is it after the learning phase that the rats begin to say, okay, I can predict now, I understand what you asked me? And no, it was also the case during the learning process. They were predicting and having this behavior. Okay, so for the discussion, so you remember that we had a particular question at the beginning about the cognitive model. We were thinking of the rat. We were not sure about what was this model. So basically it is hybrid 3 or hybrid 2. And also we were not sure about, okay, what were the states and actions that rats are taking. And you can see that here, when the rats start in one state, after it will take some, for example, a turn action to go to the next state. So we have now a precise way to describe what is doing the rat and what is the best model we can infer about his cognitive model. Definitely for pass hybrid 1 and hybrid 4, they are not good candidates. But hybrid 2 and hybrid 3 are good candidates, but they are close candidates. And so this closeness can be seen also in the negative likelihood values, which were very close for differentiating hybrid 2 and hybrid 3. But still, as I told you before, hybrid 2 and hybrid 3 have different behaviors. For example, for the incorrect choices. Some, for example, for hybrid 3, will go directly from one feeder to the other, what we call the shortcut path. Here there is a choice of the animal. In hybrid 2, it will stop and, okay, think, do I turn right or do I go straight? And finally, I would say that through Arca it is intuitive in the sense that these different segments will have less credit with respect to all the credits of the different actions the rat can take. And in the data, we can see that in hybrid 2, there are long, such wrong trajectories of paths. So, okay. Quantitatively, we could say that hybrid 2 and hybrid 3 are close, but in a sense, they are still different. And a last interpretation we are discussing with Ingrid is about, let's say that now we take a cognitive cost as the length of the different trajectories or paths. And after, let's take the benefit as a performance, the number of, in terms of the number of errors that the rat is doing. And if we look at, for example, the path model with the very long path, definitely it will have a high cost. But we saw also in the data that it is leading to less errors. So the benefit is interesting, but the cost is high. In hybrid 2 and 3, what is interesting is that the cost is less. And so definitely, but the benefit is not so much. So it seems that, and it is also confirmed by the neuronal code, it seems that the rat is kind of lazy in the sense that, okay, I don't need when I just leave this let feeder to predict the whole path I'm doing. It costs me a lot, and maybe it doesn't bring me so much. So I take my time, and at the next intersection, okay, now I can predict where I am going. And so they seem to minimize this cost-benefit ratio for decision making. Okay, so these are the different perspectives and conclusions we have about the computational models. So it seems that we were able to try to find new hypotheses that are emerging with the colleagues in biology, thanks to the architecture of these different granularity and components that we were not sure about. Now we are, so there is a link between timed activity-based contribution of actions and the behavior, so of different individuals for different models. And so now we are working with another PhD student, Usama Moore, and in collaboration with Luc Henri about the theoretical model, much more simpler than this erratic behavior, more a rational agent, to try to formalize better this result. So the next step we have is now that the top-down approach seems to be okay, we would like to go in the opposite direction, taking advantage of the neuronal code in Arca to predict better what is the behavior of the animal. Okay, so I think I am almost on time. Thank you.
Questions or comments?
I thank you very much. Is it, do you hear me? Yeah. Yeah, it's red, okay. So thank you very much for this very nice talk. Actually, I have two questions, maybe naive ones. So the first one is, I was wondering, how do you define, for instance, the action of the rat if he goes one direction, but before reaching one point, he goes back again? For instance, what happens in this case?
In the same arm?
Yeah, for instance, how do you define it?
Maybe you can see from the experiments we were discussing.
Yeah, from the experiment actually, because the alley are very narrow, so it's up at this level. So that means the animal can't really turn back. Turn back. So it just means that even if he's going in incorrect, he continues.
And sometimes you can see that the animals actually go back to the reward. And that's why we took the reward like the 0.0, because it seems that he has to go back to the reward to start again, to refresh. And as soon as he's back to this reward, it seems better. By the way, I would like to ask you, it was clear after... okay. Yeah, Alex, if you want to add something.
Oh.
You had another question, right?
If the answer is okay for you.
Yeah, so the second question is when you were speaking about this neuronal code analysis that you did to know what was happening, and then you mentioned 3% was coding for when they were in the path, and the 22 when they were at the center, right?
Yep.
I just wonder what happens with all the other with the remaining neurons. With I mean, because you mentioned, yeah, what happens if you have an idea what they were doing, what is going on?
Yeah, because it's a region of the brain that kind of messy, you know, if you can say. They're coding for many things, yeah.
So something that I didn't mention is where we plant the electrode, because we put the electrode in the striatum. So is part of brain that is involved in motor activity, but also in task learning, in goal-directed activity, and also you have two parts in the striatum, and so one taking the relay in the habit process, and that we are not showing today, but so it's work with Patricia, is she dissecate the difference between these two parts of the striatum, and we can see the very interesting relation between, okay, so when the animal is thinking during his learning process is one part, and when it's coming like an habit, because at the end of the experiment, when the animal is on the plateau, so he's really like a machine. He's doing that, alternate, and never stop. So, and he's like an habit. So I don't know if I answered to your question, but yeah.
There was one here.
Etienne, je crois, ouais. Peut-être là aussi.
Thank you. Thank you for this very nice talk. I have only stupid, naive questions.
Oh.
What is your assumption about the temporal correlations in order to evaluate the mechanism?
At behavioral level, or?
In fact, I have a lot of questions. Did you try to evaluate if when you change the animal, the decision rule is completely different? I don't know very well mice and rats, but I know humans, and I know that the same remark has not the same effect on everyone. So my question is related to, is it the same for the rats?
You mean at temporal level, or is there an individual variability between?
Yes, between animals, or depending on the time. If it's too early, it's not necessarily good to find the reward.
So fine. I would let maybe Ingrid speaking about the results in experiments with the animals, but I would say that starting the variability between the animals, we were thinking maybe it was more clear that there was a difference, but we didn't know because we didn't even know the models of the animals. But finally, with these quite different models, we notice that most of them are using a particular model which is telling something. And I would say that the difference in, from a computational point of view, the differences are quite subtle differences, in the sense that, as I said, okay, maybe the behavior for correct path is the same, but for incorrect path, it seems that the two rats are, one rat particularly over 6 is thinking differently. But I would say it's quite hard at this point to really say that they think really differently from my point of view, but maybe the biological interpretation is different, I don't know.
Okay, to complete the Alex's answer, first, you remember when Alex showed you the learning curve, you have these erratic things. So one part of the answer to explain this is because it's successive day. So you probably has a forgetting between day. That could explain this drop. So this is the first answer. But I was surprised actually that all the rats were fitting to hybrid tree, because I was thinking that they have all different learning process. I was expecting, like us, that we have different, just following, and so they don't care and they don't try to understand. And yes, I was quite surprised, but when you have a look on the learning curve also, you see that maybe the first one is more clear, but the tendency is, yeah, seems almost the same for each animal. No, we don't have all the different, sorry, it's in another graph. It's another plot that's not here. To answer to your temporal question, something that I didn't mention is taking few seconds to do the, you know more than because you have seen the 3, 4. Yes, so it's really, really fast. And so I don't know if it's answering to all your questions that you have, but yes, in terms of temporal things, so it does mean that probably you have many processes. I don't know if as we say, cognitive processes are superimposed. So it's quite in terms of the action potential also, because so it depends of the type of the action potential. So no, sorry.
Okay, another question here. After that, there are questions on YouTube too.
Hello, thank you very much. I have two questions. I would like to know if the animal was food or water deprived.
Sorry?
If the animal was food or water deprived. It was? I have a second there.
People on Zoom cannot hear. No, but people on Zoom cannot.
Sorry, just to motivate the animals, yes, we deprive a little bit. So, but you know, usually that we are doing is depriving the animals, and we don't leave the animals go under 90% of his own weight, the original weight. So, but it's just a little bit deprived to be motivated. So that's the first thing. No water deprived, not at all. Second question.
Is it possible to differentiate an exploratory behavior, the reward behavior, and the decision making, just looking for the neurocode? Yeah, because you tried to identify which neurons are coding for decision making, right? You try to look which neurons are deciding which direction to take in the maze. Okay.
Sorry, Yuri. I want to be sure to understand your question. Your question was, can you dissociate decision-making process from motivation? Is that? From exploratory behavior? Definitely. Definitely, and I'm sure that is a combination of the both.
Yeah,
I don't know.
Yes, but from my point of view, with what we have, we cannot differentiate, in the sense that this erratic behavior, when we discovered it, we had many fancy hypotheses discussing with the biologists. And finally, they said, okay, but it seems that the rat is bored the whole day, And when you put it in the maze, it is like us. Okay, he wants to explore, but how to differentiate the exploration process, which is already a complex process at the cognitive level, with respect to other process? With the tools we have here, we cannot. So
just something to add. It's true that it was 20 minutes. We put the animals 20 minutes in the maze. So that means it can do, I mean, we have seen some rats who have done 100 paths, something like that. So it's a lot. So as you say, we can't really dissociate the exploration and boring phase from the...
It doesn't explain everything, yes. Yeah.
If the cage of the mouse is connected to the maze, when it's bored, it can just leave it.
Yeah, so we want to go home.
You can just let the camera there.
Because they have the cable.
Ah, okay. Maybe if it's Wi-Fi.
You are used to, huh?
Okay. Okay. So thank you for the talk. I have two questions. In fact, one a question, a comment. First, this is a question related to exploratory phase. I think it also depends on with which species you're working with. I don't know. I'm not a biologist, but I would expect that, for example, a human, maybe not exactly the same experiment, but an experiment based on making some choices between two options and get a reward and try to maximize the reward. Sometimes humans, at least, this is what we observed, even if they show that they learned based on their performances, sometimes they really just they just want to play, they just want to see, because they don't know what exactly the task is, and they just say, okay, apparently I do good performance the last time, but what happens if I change my mind? And I'm not sure how to quantify such a behavior afterwards. That was not a question, but rather a comment, which I remembered from some other things, some other studies. My question is that it's maybe not really pertinent to that context, but did you ever try, or do you have any plans to try also changing the trajectory even after the animal learns the task, to change on the road and to see what happens next? Or what would you expect to observe if it was not done? In the case of mice, rat.
Yeah, maybe Ingrid is more adapted. I know that they have another experiment where sometimes they're providing the food with some probability.
Is that I would like to talk.
Okay.
We changed the probability between, because so the role here was, okay, you have to go to the right, you have the palette, and go after to the left. And we checked the percentage of probability on each. Yet we haven't analyzed these data, but so I think, as you said, because you said something very interesting about the animals just want to try a new path. And we can see that even if when they are on the plateau, they still have some erratic behavior, like, oh, now I'm feeding off, or I just want to explore, or do something. So I think you still have that. And when if we change the probability, that I guess, from my biologic point, we are going back to a goal-directed process where you have to learn a new role. And I think it will re-engage probably some neuron that we have already shown to be involved in this goal-directed process.
You change the task to learn.
Yeah, and that is very interesting in terms of the flexibility of the behavior. Is that we label in biology the flexibility of the behavior, because it's something that is really relevant between normal cognitive process and pathological cognitive process that we can have.
Okay, so just quickly, the two questions on the internet, because they are waiting for a long time. The first is for Cyril, who asked how we choose the model. Is it AIC, BIC, etc.? And since this is for me, we did hold-out procedure. And if you want more detail, you will have to see the preprint once it's released, because we have not done it yet, but soon. And then you have two questions from Claudia, who said, excellent conference, thank you very much. How to explain how does the model take into account the high behavioral variability even after the rat has actually learned the task, which is more or less what you did, but that's the first question of Claudia, and then there is another one.
So it is how to explain after the learning process
how
that there
to
is
take
a
into
difference.
account the high variability, or how the model explains the high variability after the learning phase.
Oh, that's a nice question, interesting question. So,
in terms of behavior, Claudia, I think I already partially answered to this question with the forgetting overnight because it's a different day. But it's true that in terms of the model, is it the gamma with the forgetting?
Yeah, maybe. Yeah, but I would say that we represent. I would say quantitatively, we are close to the curves as you can see. Now, what knowledge we can infer from that? Yeah, I would say that it is related to Etienne's question and your question that we have this notion of, okay, we can see that the process is really dynamic because here it's even abstracted as the notion of session, so during whole day. But there is this notion of, okay, is it that the rat is increasing the credit of different actions inside an experiment? And is it that the rat is, okay, I don't like to call it maybe forgetting, but maybe forgetting something in between the sessions. So it is, I think, why we are closer to the curves. Now, as a question of interpretation, do we show, I would say for all the individuals, we show that there is this variability, we can model it. Now, how to interpret that, I don't know personally.
Okay. And so the last question from Claudia, do you expect to compare statistically the neuronal activity retrieved at each model step? I think it's more for me than well, we have, even the analysis that we did was a bit tricky because we need to compare a good path with loop. And so since in some sessions there is no loop, we cannot make this computation. So it means that we need to compare paths that are often there. So of course, because of the learning rule, we can compare the two good paths. This is very easy. And since they are common only in the step SEM, we did it, but it's very high, the difference for sure. But this could be interpreted as they remember from what was the last feeder, they remember just where they go. So it's hard to interpret what means this difference between those two good paths. And then if we want to compare good path with something else and have pieces of paths that are common to both sides, well, we are a bit stuck with the loop. And that's where we just stop there. Because after that, all the difference you can make, if they are not in common portion of the maze, they may be interpreted like place cell because this is just they are not at the same point, and it doesn't mean that the rat really knows what he wants to do. So because of all that, I'm not sure we will be able to make much more comparison than that. We are thinking to look more precisely at the choice point on the bottom, because we looked at just the two branches, and maybe there is something on more precisely at the bottom, but that's a little bit the kind of extent we can have a look at.
Yeah, I would say that it is more the results and the framework we had that converged to these questions, because otherwise testing exhaustively all the possibilities. I mean, when we didn't get this result of Arca at the cognitive level, so it was some weeks ago, we didn't even think about testing this hypothesis of the rat. So, and after exploring exhaustively, it's more, I mean, it's a lot of, okay, for example, I will find something at the computational level. I will ask Ingrid, okay, does it make sense to you? And after we try maybe to modify, and after I said, okay, Patricia, I know there are very few data, so can you have a model of that hypothesis? And I can feel it. It's more like a guide of interaction going like the modeling and simulation
being
process.
Being exhaustive, in my opinion, statistically, it's not possible because there are too few paths of each kind to make the full, complete comparison between all of them. That's for sure. Okay, I think we are a bit late, and so thank you so much.
I didn't say, but I would like to thank the organizer. Thank you, Patricia.
And so now it's the turn of Uri Eden. Oh yes, I have to switch. Sorry. So I think now is the turn of Uri Eden. I don't know why we don't see him.
Can you hear me?
Yes, Uri is here. So Uri Eden, so it's a pleasure to welcome you. You're in the United States. We have all the countries that are represented today. So thank you so much for taking time to give this talk. And so the floor is yours, and you will speak about point processes modeling. Thank you so much.
Great, thank you. Can everyone hear me?
Yes, we hear you. Thank you.
Okay, fantastic. So it's a pleasure to be speaking with all of you. I really wish I could be there, and I hope that there'll be lots of opportunities in the near future for me to come and visit.
Wait a minute, just it's a bit low. I don't know if they... try again, please.
Can you hear me? I can also put in some headphones if that helps a little bit. Should I try that?
I don't know. Do you?
Okay, let me try it.
It's just that I think it's okay for the Zoom, but in the room it's a bit low. I'm not sure.
Is this a little bit better?
Oh yes, perfect. Thank you so much.
Great. No worries.
So your turn.
Okay, so today I wanted to talk a little bit about some of the statistical challenges that arise in modern neuroelectrophysiological experiments, and some new approaches that we've been developing to try to deal with those challenges. And in particular, we've been developing methods related to the theory of marked point processes to understand, instead of how individual neurons code for information about the outside world, how populations can simultaneously code. And I want to frame this in terms of a case study trying to understand hippocampal replay and see how the types of statistical challenges that arise in the analysis of hippocampal replay and some of the methods we've used to try to overcome those challenges. And so thanks to Ingrid's very nice talk earlier, I don't need to explain, action potentials and how they relate to local field potentials, but maybe I can just mention that modern neuroscience experiments often require a lot more integrating of information across multiple sources. Instead of a very simple stimulus response paradigm where we look at the activity of just a single neuron in response to a stimulus or to behavior, oftentimes now we're recording from hundreds to thousands of neurons simultaneously and trying to understand relationships between the activity of all those neurons, potentially in multiple brain areas, to rhythms that are recorded in the local field potentials that integrate synaptic potentials across thousands to hundreds of thousands of neurons and relate those all to features of behavior. And this reflects I think a larger trend in neuroelectrophysiological experiments of increasing complexity, both in terms of the stimuli, the response, and the associations that we're looking to understand between them. In particular, in terms of the stimulus, we've moved largely from simple stimuli that were entirely experimental controlled, typically one-dimensional, to ones that are very high dimensional, that are quite naturalistic, that an animal might experience in its actual experiences, that are largely uncontrolled within the experiment. And we've moved from responses that typically look at the activity of an individual neuron to responses that look at the coordinated activity from large populations of neurons and how those relate to various rhythms in the brain. And so we've also gone from looking at a single type of modality, maybe just action potentials, to responses in terms of multimodal signals. And more than any of that, we've moved to trying to understand more complex associations. For example, from understanding stimulus-response relationships that we typically assume to be stationary to ones that explicitly try to capture dynamics in the representations. And in particular, we've also moved from trying to understand directly observable relationships where what's being encoded is entirely controlled and observed to ones that reflect maybe neural representations of cognitive features which can't be directly observed and must be modeled using some type of latent processes. So I wanted to discuss the ways we've dealt with some of these challenges in the context of a particular case study, trying to understand hippocampal replay in awake rats. In particular, if you have a rat exploring an environment and you record from the medial temporal lobe, in particular the hippocampus, people are largely familiar with the notion of place cells, where if, say, this rat here moves from the blue area through this green area to this red area, and you record from multiple neurons simultaneously, you may record neurons with place fields that are more in the blue area that will respond when the rat is moving through that blue area. Other cells with place fields in the green area, which won't respond when the animal's here, but will respond when the animal starts to move through the green area. And then maybe other cells with place fields in the red area over here, which will respond only when the animal moves through there. And so as the animal moves through its environment, what you can see is a very active rhythm in the local field potential, in this case, a theta rhythm, that corresponds with this place-specific coding. And if you look at the activity over this population, you see a pattern of spiking moving from these blue neurons through the green towards the yellow and red. And all of that is related to the behavior in terms of moving through this environment. But it's been long seen that when the animal isn't actively exploring, when it's sleeping, or even when it's awake and at rest, either before moving through these environments or after moving through these environments, what you can see as shown in this kind of red box right here and expanded right here is that occasionally there'll be a change in the local field potentials reflecting a sharp wave and a fast high frequency ripple event. And when that occurs, you'll see patterns that have features that are statistically similar to when the animal is moving through the environment. In this case, the animal is stationary at this point, but you see a pattern of the neuron that fires with the blue receptive field firing first, then the green, then the orange and red. And so you see similar patterns, which are temporally compressed, but don't just represent temporal compressions of the data itself. They match in some ways statistical features as the animal moves through this environment. And similarly, we can see another example here in blue, where you have, again, this high frequency ripple activity. And now you see activity which doesn't actually reflect anything that you would see during actual experiments. In this case, the red neuron fires first, then the green, then the blue. But again, you have a pattern of activity that's in some way related to the population pattern that you see during active exploration. And so our goal is to try to understand these various types of replay events. In particular, our experimental collaborators, in particular Lauren Frank's lab at UCSF, has already shown that if you interrupt all of these replay events with stimulation, then the animal has trouble learning new navigational tasks. In this case, the animal is moving on an M-shaped maze. It has to alternate from the center left and then right, as we saw in the previous talk. And if you interrupt all these replay events, the actual behavior itself never reaches the same behavior as an unstimulated animal that's allowed to have these replay events. But our experimental collaborators were interested in these various different types of replay events and whether they could determine which types of replay events were going and stimulate them based on their actual content. And so this presents a number of different statistical challenges that I wanted to take some time to go through. Maybe the first and foremost is they want to be able to detect the content of these replay events in real time. And so that means that they don't have time for the spike sorting step that was explained during the last talk. In particular, if we have maybe multiple neurons that are recorded on the same tetrode, those neurons all have separate waveforms. And various spike sorting techniques are available to maybe find various templates, in this case, maybe red, blue, and green, representing 3 different neurons here, to sort each of the spikes based on those templates, and then to build separate coding models for each of those neurons. So instead of spike sorting and building a separate neural model for each of those neurons, our goal is to try to directly relate the coding properties of each spike based on its waveform without making an explicit spike sorting step. And so the approach that we used here is that instead of using a simple univariate point process model for each neuron, we built marked point process models for entire tetrodes that may include multiple neurons, where we defined a joint mark intensity function that we call lambda here. It's changing as a function of time, but also has this multivariate mark that in this case might reflect things like the principal components of these waveforms, or maybe just the peaks of these waveforms on multiple electrodes. And we characterize the probability of seeing a spike in the neighborhood M of a particular mark, of a particular waveform at each moment in time. That's what we build the model to describe. And then we can use those models to try to decode the animal's either position in this environment or these replay events by looking at sequences of spikes with particular waveforms. And so here's just an example of the difference between these 2 approaches. So on the top is just the classic approach, looking at an individual neuron and trying to build a receptive field model. In this case, it's a spline-based receptive field model, a spline-based pace field model, where we've linearized this M-shaped track, where this origin, this O point here, is the center, and trajectories that move from the center to the right are located to the right of that origin. Ones that move from the center to the left are to the left of that origin. And if you spike sort one of these neurons, you find a place field that tends to fire just before this A location as the animal moves up the center stem. And it looks like it fires more when the animal moves toward the right over here than when the animal moves towards the left. And you can conduct a test, and in this case, for that neuron, you'll find statistically significant differences between right turns and left turns. However, if we build a joint mark intensity model, in this case with the mark just being the peak height of each spike, what we see is that when the peak height is very large, we get the neuron firing with spikes that look like that about equally before turns to the left and turns to the right. But we also have some spikes with slightly lower magnitudes, slightly lower peaks, which can't be distinguished, can't be separated by spike sorting from the ones with larger ones, but the ones with the smaller peaks tend to only fire when the animal is moving to the right. And so in this case, if you actually saw the waveform itself and you had this joint mark intensity model, then you'd be able to tell if you saw maybe a spike with a lower amplitude, you'd be able to predict with higher confidence that the animal was about to take a right turn. Now, maybe going back to looking at higher dimensional marks, so in this case, looking at the peak heights from each of 4 electrodes in this tetrode, what we see is that if we build one of these joint mark intensity models, we capture simultaneously both features of the spike sorting. So in this example, I'm showing, for example, the peak height in channel 1 versus the peak height in channel 3, and the intensity, just a slice of the joint mark intensity as a function of channel maxima. And you can see that the same information that goes into spike sorting is present in this joint mark intensity model. While simultaneously, if we look at, for example, this linearized position as a function of channel maxima, then we see individual blobs that represent the place fields of each of these kind of clusters. But now one of these models is capturing these multiple place fields simultaneously. And we have multiple mathematical approaches for capturing these types of joint mark intensity models. For example, we can build non-parametric models that use kind of kernel-based estimators that just place a maybe Gaussian kernel at each observed spike location both in terms of position, but also in terms of the marks. Or we can build a parametric or semi-parametric model, say as a mixture of Gaussians, that tries to capture each of these individual blobs with its own Gaussian structure. Okay, so that addresses potentially the first challenge of not having time to spike sort. A second challenge is that in the experiments, these representations tend to be dynamic, and they tend to be dynamic in multiple ways. The receptive fields of each neuron tend to shift. So for example, the shape of a place field as a function of position is known to change as a function of experience. These place fields often tend to become more narrow, so become more place specific. They tend to shift in their centers, and they tend to increase in their firing intensity with experience. But in addition, you may have changes as a function of marks. For example, you may have electrodrift, which leads to the spike waveforms changing through time, or you may have new marks, new clusters of marks appearing or ones disappearing as neurons die or are electrically revealed to the electrodes. And so one of the statistical challenges is how to, instead of treating these models as stationary models, track their evolution through time. And we've come up with a number of approaches, including some simple ones, such as just including the spike times themselves as an additional mark in the model, to an approach that I'll discuss a little bit in a slightly different form later, using state space models to actually explicitly model temporal changes in these clusters. But another maybe simple way that I want to briefly discuss is a hierarchical modeling procedure where we use a Gibbs sampling method to actually track the evolution of each of these clusters through time. So in particular, this Gibbs tracker approach consists of two steps. So we're using our mixture of Gaussians model that I described previously. The first step is a stochastic cluster assignment where we take each spike that we observe and we, in multiple iterations, assign that spike to one of the clusters in our joint mark intensity model. And then in each of those iterations, we take all of the spikes that are assigned to that cluster and use a standard Gibbs procedure to update both the center of the model in the mark and position space, the span of the model in mark and position space, and the intensity of that component of the model. And so that can allow us to track all of those changes through time. And so I can just show you an example. So this is an animal moving through a circular maze. I'm just showing a linearized position here on the top plot, and then the peak height in terms of the first and third tetrodes, and then just snapshots of those. And then in different epochs, so the epochs are marked by these red bars here, we've tried to build these joint mark intensity models using a mixture of Gaussians approach. And what you can see, if you go from this green epoch to the cyan epoch to the red epoch, if you just look at the activity and the spike marks and locations in time, it's very difficult to just see by eye changes in the population activity. But if we look at our model fits, both as a function of the joint mark intensity across these tetrodes and as a function of position, what we can see is the expansion or maybe growth of a new component that represents activity at position, maybe at about position 1 here, where we have a very small component with quite large amplitude spikes in terms of at least tetrode 3, which then expands, maybe shifts its center and expands to a broader range of spike waveforms. You know, whether that's the appearance of a new neuron or whether that's one neuron's receptive field and space of waveforms expanding, we can't say, but we're able to capture that transition using this approach. A third statistical challenge when we build all these models is how we actually determine whether or not the models we've built are accurately capturing the statistical properties of spiking, the coding properties of the neuron. And in particular, with these kind of high dimensional models, it's very easy to get models that seem like they're capturing a lot of the structure and the activity, and yet may have a lack of fit in various components. And so we've expanded upon traditionally known point process goodness of fit methods, which use time rescaling to assess the quality of fit, for our joint mark intensity models, which use a combination of both time and mark rescaling. And we've come up with a number of different transformations. In this case, I'm showing one of those, which is a joint mark and time scaling. So in this case, the SIs are the actual spike times, the MIs are the spike waveforms for each of those spikes, and we're transforming those into a set of UIs and VIs, where the UIs integrate the joint mark intensity for each observed spike waveform up to that observed spike, and then normalize it. And then we also rescale all the marks and get a distribution of all the waveforms that we have, and then use a Rosenblatt transformation for each dimension of the mark in order to get a transformation where, if our joint mark intensity is correct, our spike times and spike waveforms should transform into these V's and U's, which should be samples from a uniform distribution in a D+1 dimensional hypercube, where D is the dimension of the mark and the extra 1 is the dimension of position in this case here. And so this is just an example for some of these models that I'm showing, where we have a joint mark intensity as a function of a linearized position here. But you actually have a drift in the marks through time. This is, if you sort these, you actually get activity from 2 neurons here, where both of their spike waveforms in one dimension, in this one-dimensional mark space, are drifting through time. And if you don't have a model that captures that drift through time, you can see through this transformation, that misspecification, in the way that these spikes are transformed and are not, and you can use simple known statistical tests for uniformity in the cube to reject the hypothesis that these are uniform samples, and therefore reject this joint mark intensity. Whereas if we build the type of adaptive model that I just described and then perform this transformation, then at least using this transformation, you end up with samples that are not distinguishable from a uniform. Okay, so that's the third challenge. The final challenge I want to discuss is how you actually invert this process of building the model in order to decode either the animal's position through time, or in terms of these replay events, to decode the animal's cognitive state as it maybe perhaps mentally explores positions in this compressed time frame. And so the way we do that is we use these joint mark intensity models to describe the relationship between a cognitive state process that I'll describe as X here, which describes either where the animal is in its environment, or for these replay events is a state that represents maybe continuous cognitive paths through the environment. And so the models that I just described define a relationship between these states and a set of point process observations. Our Y's here are our spikes and the waveforms. And we add to that another state process, in this case, a discrete state process, which I'm defining I1, I2, I3, which actually not only controls the observations and how they change in time, but also controls the dynamical model of this continuous state. So for example, if we want to detect whether or not our neuron is decoding actual position versus replay, or whether that replay is a forward replay or a reverse replay, or a replay of an outgoing trajectory versus an ingoing trajectory, we can code all of that with our discrete state process. And then once we've defined that, then the mathematics for actually decoding ends up being quite simple. If we want to compute the joint distribution of our discrete state and our continuous state as a function of the increments of our point process and their spike waveforms through time, we can express that posterior distribution at each moment in terms of that same posterior distribution at the previous time, multiplied by a state model that describes how our state processes evolve through time, how the animal either moves or how it cognitively explores, with our joint mark intensity model that describes the probability of observing a spike with a certain waveform as a function of our discrete state and our continuous state variables. And so we can compute that moment by moment. And this is one example of just a decoding of that discrete state. So in this case, the black is the animal's actual locations. This is all actually very quick. It's about a quarter of a second, this whole movie. And what I'm decoding here, in red, I'm showing just the maximum a posterior estimate. And the color code around that is showing the posterior distribution of this cognitive state through time. And so what you can see is the animal's not moving at all, but in this quarter second, we have evidence in the spiking of a trajectory, a replay trajectory that moves away from the animal's location, takes a left turn, and moves up the track as well. In addition to estimating this kind of mental cognitive state, so I'm showing here just a snapshot of that. So this is one replay event. Again, this linearized position from O, the center, either left or right. And now I'm showing that posterior distribution linearized through time. But in addition to estimating that continuous state, we can also estimate a discrete state. In this case, the discrete state takes on one of four possible discrete states. It's either an outbound or an inbound replay event, and it's either a forward replay event or a reverse replay event. So it's either looking at a movement or a mental exploration of the movement in fast forward, going forward, or rewinded. And what we can see is that if we're estimating this x variable, the position, simultaneously with the state variable, after just a few milliseconds, in this case within 100 milliseconds, we're able to determine with a high degree of confidence the type of replay event this is, as well as the trajectory of the replay event. In this case, I'm showing two replay events, both of which start at the center and move outward, either to the right or to the left. But in fact, these are quite different. This first replay event represents an outbound forward replay event. So the animal perhaps thinking about moving away from where it is in fast forward, versus this replay event, which also moves away, but is actually an inbound reverse. So the animal replaying its movement from the outside, from the left back into the center, but then that movie played in rewind. And so this gives us our first steps towards potential closed loop interruption. Our experimental colleagues at UCSF have given us the challenge of trying to, within 50 to 100 milliseconds, understand the content of each replay event so that they can potentially stimulate and interfere with these replays within that, time period. And so if I require a 80% confidence in my discrete state, then in fact, for nearly all of the dozens of replay events in a particular experiment, I'm able to determine which of the four discrete states it has within 100 milliseconds. And the more confident I need to become, the longer it takes me to become that confident. And in black here is the fraction of replay events which I'm not able to detect with confidence. But this shows us that we're able to move using all of these approaches towards potential closed loop interruption. Okay, so I've tried to highlight a number of different statistical challenges here and give a taste of the way in which these marked point process modeling approaches can help us address each of those challenges. And in particular, these marked point processes allow us to understand coding properties of large-scale populations, assess the qualities of those models, and infer features or decode features about the signals driving the neurons. And this state space approach that I described in this fourth challenge provides the kind of link between either stimuli or behavior and the neural activity, or in a case like this, between complex cognitive processes and neural activity, which gives us the potential for these types of closed loop experiments where we're using information inferred moment by moment from the brain in order to drive our experiments. So I'll end there. I just want to acknowledge the many people who helped devise these algorithms, who actually implemented them, as well as my funding sources. Thank you very much.
Thank you a lot. Thank you, Yuri. Uri, sorry. Yeah, Yuri is in the room. I'm sorry. Who has a question or a comment? Yeah, Ingrid, go ahead.
Sorry. Thank you very much, Uri. I'm sorry because I didn't really hear properly, so I would like to be sure to understand from the biologist's point. Tell me, with the tools, you label Gibbs, it was something G-I-B-B?
Gibbs sampling.
Yeah. Does it mean that we can affirm that we are recording along the session the same neuron?
Yeah, so that's a good point. You know, if over a long period of time, something, you know, you're looking at your neural activity and it looks like it's changed through time, you know, either maybe through different epochs of the same experiment or, you know, over multiple days, there's the question of how confident can you be that you're recording from the same population? And, you know, if, for example, I look at the data set from here and I look at the end, you know, the beginning of this experiment and the end of this experiment, you know, it seems like I have some place fields here that look about the same. So maybe this neuron here, maybe this cluster represents a single neuron here, which looks like it's the same at the beginning and the end. What this Gibbs sampling procedure allows us to do, as well as some of the other approaches that we've taken to address this problem, is it allows us to track moment by moment how these are changing. And so one consequence of that is you might be able to see maybe a change from the beginning to the end or a change from one day to the next, and you might be able to track that change through time. And so there is the potential here to be able to say that, you know, this cluster that's changed a little bit in its coding properties and maybe even changed in terms of the shape of its spike waveforms, because of the kind of continuity of that change through the experiment, I can say with high confidence that it's still the same neuron and it's just become more specific with experience. Or to counter that, I may be able to detect a sudden shift, a sudden change between this, and I might be able to infer from that that in fact this is a new neuron that suddenly has appeared that has coding properties maybe in a similar area to this previous neuron. So I'm not sure that I have an answer that you can always tell whether or not you're doing this, but I think these approaches give us new ways to potentially assess whether or not you're recording the same neuron and whether its receptive field properties are changing through time.
Cool, thank you very much. I think you solve a huge problem in biology because so I would like to ask you the question. All the time the referee tell us, okay, because we have to move the electrode, because that we didn't mention to the public here is we can't leave the electrode on the same place because we have a glial cell around. So that means we have to move the electrode. And sometime when I'm moving the electrode, I still have the same signature of the neuron, but I can't assume that is the same. And so the referee all the time told me, you can't say that you follow the same neuron.
Yeah.
I think it's possible with your tools.
Yeah, so it might be possible to track it. At the very least, what you could say is you could say this neural population is coding with the same properties. And so, you know, even if I don't know that, say, this neuron here and this neuron here are the same, I can say that the population as a whole still seems to have a neuron with, you know, similar waveform shapes that's coding in the same way as it was before. And so if I'm discussing it at the population level, I can talk about kind of maintenance of information at the population level. You know, there's maybe potentially another step, but there's potential here in also saying, well, in fact, I'm confident that this was a neuron and that neuron has just shifted through time.
Thank you a lot. Other questions or comments? I have a few because it was really, really interesting. First of all, so I understand that in the end you want this closed loop detection, which means that you already know the kind of path the rat will be taking in the maze, so you know already the succession that you want to see, and this helps you to predict who is going to code for what. But if you say you don't know the maze, or you kind of try to guess what is the succession that the rat is making, could you kind of infer also the succession of the place on top of everything else? Or you really need to know in which seconds they are going to be?
Yeah, so in general, we've applied these types of methods in less structured environments. So in open field environments, or in each of the experiments I'm showing here, these rats are very well trained. And so the receptive field structure isn't changing very much. Or the hope was it wouldn't change very much, even though we found these changes. We've also applied these in kind of open field, less structured tasks, and when the animal is first introduced to these types of environments. And in those cases, we always need the ability to build a receptive field model, in this case, a population receptive field model that relates things like the animal's position and the spike waveforms to the timing of events. But in those cases, we have been able to use these methods to, say, decode with high accuracy the animal's movement through an environment. It's really this structured of alternating left, right. What that really helps us with is in these replay events to be able to disentangle these kind of leftward versus rightward trajectories. And in the cases where we're trying to predict actual movements, we're able to use them to predict right and left turns before the animal actually makes a turn. And so I think these methods as a whole are largely applicable even in less structured environments and less structured tasks, though in particular, this selection between outbound and inbound really relied on the structure of the task. Another thing we've done in the less structured task is try to determine whether or not these replay trajectories are coherent, whether they represent continuous mental exploration, or whether they're more fractured, whether they jump between environments. And that, again, doesn't require as much structure, this type of left-right alternation in this task.
Okay, thank you so much. And another question for maybe something which would be much more general than hippocampus and position coding. Would you be able to incorporate to the spike sorting other relation, like, for instance, connectivity between the neurons that would help you to decode, well, to spike sort the whole thing by adding those correlations between the spike trains or stuff like that?
Yeah, that's a great question. So, over the past 10, 15 years or so, classic univariate or multivariate point process models looking at multiple single units that are spike sorted have done a lot of work trying to understand connectivity between neurons and how using point process models can help you understand, pairwise relationships between neurons and how those adapt through experiments. We believe the same is possible with these joint mark intensity models, but we haven't actually implemented them yet. There's an additional challenge in trying to figure out how to represent, if you're not separately spike sorting, how to represent correlation structures between these types of clusters. So we can imagine what it would look like. So you can, for example, look at one of these clusters here and imagine that each of these kind of blobs represents a neuron, and how a spike in one of these blobs would then decrease the intensity in its own blob temporarily because of the refractory period, and maybe with some time lag increase or decrease the intensity of some of these other blobs. So it's certainly possible. It's actually an area of active exploration right now, but we haven't implemented those yet. But I think there's a lot of potential in adapting these joint mark intensity models to have that type of correlation structure.
Okay, thank you so much. Other questions or comments in the room? On internet, I don't think so. So thank you so much, Henri, for this beautiful talk. And
thank you very much.
We have a half-hour break right now, and then we will come back for Thibaut Taillefumier and Robert Cass. Thank you. very soon. Could you just try to talk so that we see if we hear you well?
Hi, can you hear me?
You're talking?
Yes, I'm talking.
Okay, we have a problem. Don't wait, I will see with the people upstairs. Tu vas lui dire? Oui. Ben on l'entend pas du tout.
On l'entend pas dans la salle.
Ouais. Sorry Thibaut.
That's fine.
Can you try again?
Yes. How are you?
No, we don't hear you. Wait. On n'entend rien? Si? Can you just try to speak again?
Allô, allô. Allô.
Yeah. Parle en continu, s'il te plaît, juste pour qu'on arrive à régler le son au fur et à mesure.
1, 2, 3. 1, 2,
Désolé.
1, 2, 3. 1, 2, 3. 1, 2, 3. Allô, allô. Allô. Allô.
Please, everyone,
Allô.
just we try to make something with the sound.
Allô.
Continue to talk.
Allô, allô, allô. Allô.
C'est un peu mieux, mais... Hein?
Allô.
Yeah, c'est encore plus bas que pour Eden, non?
Ah ouais, mais déjà, Yuri, déjà, on n'entendait pas grand-chose.
Ouri. Oui. En fait, il faut lui dire que le son est trop bas dans la salle. Que c'est encore plus bas que pour Ouri. Oui, il faut que Thibaut parle pour qu'on arrive
Allô. Allô. Allô.
Oui, je sais pas, lis un truc, on continue. Pour qu'on arrive à régler au fur et à mesure.
Je sais pas quoi dire. Allô. J'ai de l'écho. C'est normal?
T'as de l'écho?
Ouais, parce que je sais pas. Ok, je vais couper mon son peut-être.
Non, moi j'ai coupé tout.
Ok, ok. Vous m'entendez?
Oui, on t'entend.
Ok. All right.
On va commencer. Donc, je vérifie que là-haut c'est bon. C'est Elios? Je sais pas. Oui, super. Donc, on a le plaisir d'écouter maintenant Thibaut Taillefumier de l'Université du Texas, qui va nous parler de réplica Minfield. Merci beaucoup Thibaut.
Ok, donc vous m'entendez, c'est bon?
Oui, on t'entend.
Okay. Okay, I'm gonna switch to English. So thanks for the invitation. Apologies for not being there in person. I wish I could have been there actually. It's my birthday today. It would have been great if I had been in Paris, but whatever. All right. So I'm going to talk about replica mean field as you were just told. So that's going to be a little bit different than the previous talk because it's going to be about modeling. And basically, it's a point process-based modeling framework that I've been developing in close collaboration with François Bacheli. And we've been working on this for about 5 years now. And so I'm going to give you like kind of an overview of the method. And I know that on the program, there'll be talks that are going to be very closely related to that framework. So I hope it's going to be a good introduction. So, okay. So basically, this slide is just basically a word of caution when you do modeling in neuroscience, and it's that, okay, it's a big task, and the system you're trying to model are gigantic. They have many different constituents, and all those constituents display tremendous heterogeneity. So, so when you do modeling, you want to be humble, and you know that you're going to have to do a drastic simplifying assumption, and in doing so, you're going to throw away a lot of details, but okay, you have to do it anyway. So I'm just quickly introduce the kind of simplifying assumption I'm going to make, and then I will introduce the framework. So there will be a simplifying assumption at two levels, right? The first one will be at the level of the constituent, so the neurons. And I like to exemplify the ingredients I like to keep on a movie. And basically, if the movie was playing, okay. Well, the movie is not playing. So I'm going to describe what you were supposed to see. I mean, I don't know if you guys have seen that. This is like a Japanese fountain type of thing. And basically, it's supposed to receive water, go down, release the water, go up. And so basically, that tells you that there's three ingredients to it. You want, if you think of it as a neuron, you want the neuron to continuously integrate its input, then interact with its surrounding in a punctuate fashion. So at some point, boom, you release everything. And then once you're done, you reset to a base state. So this basically memory erasure. And so you want those three ingredients, or at least I will ask that a decent neuronal model has those three features. And there's different flavor of that, right? You can have an integrated and fire neuron, you can have a rate-based neuron, and all different sort of neuron that have those three features will be rate-based in this talk, okay? So, so that's for the neuronal model. Now then, perhaps more importantly, you also need to make assumption about how those neuronal models are going to interact with one another, and that means you need to make assumption about the network. And here we're going to focus on one of the most common assumptions that allow you to do a little bit of analysis about those models, and that is the mean field approximation. So the mean field approximation is a long story. It's been proposed by a computational neuroscientist, as well as mathematician and physicists and so forth. And basically, it consists in considering a simplified dynamics that will take place on the network of neurons that have infinite size. And for this dynamics to be well posed, you also need to make an assumption about the scaling of interaction. And there's typically two scaling hypotheses. There is the classical thermodynamic mean field hypothesis, which is that a neuron have an interaction strength with one another that scale inversely proportionally to the size of the network. And then there's another assumption that is valid for balanced models. And that assumption allows for like a less stringent scaling, which would be inverse of the square root of the system. So again, there's a lot of work on this, and it's very great work if you ask me. But there's also a few limitations to this mean field approximation, and those limitations are the reason why we try to develop or elaborate a different framework. I mean, not really different. We try to extend a little bit on the mean field approximation. So what are those limitations? So the first two limitations are the ones that are really problematic. The third one, a little bit less. So the first one basically is that by design, when you make a mean field model, you throw away the correlation. And correlation may be important in the activity that you're trying to simulate. So that's a problem. The second limitation has to do with the fact that considering an infinite size system is not neutral. And in particular, it's not neutral if you want to like study the typical behavior of a system. Because looking very far in time and looking at very large system involve taking two limits that don't commute with one another. And so that means that if you look at a very big system, you may end up looking at a system that is not displaying the relevant behavior that you were interested in, because ultimately networks are finite, right? Even so they're very large. And so the last limitation is that often you assume the mean field dynamics. You don't necessarily prove that like it can be reached within a good limiting process, and that can lead to considering dynamical equations that are actually not well posed. There's no physical system that will satisfy those equations. And so, so we look at those limitations, and that was the starting point of us developing a framework, was to say, okay, can we come with a set of models that will always be well posed, and that may allow us to attack point 1 and 2. We're not saying we're going to solve it. We just hope that there was a chance that we can, like, do something about 1 and 2. Okay, so in that model that we propose is the replica mean field model. Okay, and so I'm going to explain it now on a very simplified dynamic that belongs to a class of dynamic called Galvez-Lochaber. So basically, the concept of replica mean field limit will not matter with respect to what model we're considering. I just consider this model because this is the simplest possible you can come about that display the kind of feature I required earlier in the introduction. Okay, so we call that model the counting neuron model. Okay, so here you have 3 neurons and they interact with one another via interaction parameter mu. So each neuron has its own dynamics and is characterized by a single variable, which is a counting variable. And so what is that variable counting? It's counting the number of spikes it has received from other neurons since the last time it has spiked. So here's an example of the dynamics. Each time, like, red guy is spiking, blue and green are coming up by 1 unit. And when either of those are spiking, you go down to 0, okay? And so then there's interaction between those neurons because now each neuron emits a spike in linear relationship to their count. So the more spike you've received, the more likely you're going to be to spike. And that corresponds to assigning a stochastic intensity in this fashion with a linear form. Okay? So because of the correlation, it's very hard to actually entirely characterize this system. And it may be surprising, but it's already a difficult question. So then you would like maybe to do the next best thing, which is to find a dynamics that is closely related to that system, and that will be amenable to analytical characterization. And so that's the purpose of the replica mean field approach. And that approximating dynamics is obtained as follows. So the idea is to say that, okay, I'm going to consider my system of interest, and I'm going to, like, duplicate or replicate it. And here I'm just making, like, a 4 replica model of my original system. So for the original system, whenever a neuron interacts, it will interact with the neuron it connects to. So that means that in principle, when a green guy over there would spike, it will send a spike to the guy within the same replica if I was modeling the exact dynamic. Now, the point of the replica model is that I'm not going to simulate the exact dynamic. I'm going to only simulate an approximating dynamic that I get by randomization of the interaction. So that means that when the green guy spikes, now it's still going to talk to a blue and a red guy, but it's going to pick those guys uniformly and independently across replica. And then I'm going to get the exact replica mean field limit by considering the limit of an infinite amount of replica. Okay? And so basically, this replica process is really the defining feature of the approach, right? And then there will be a different type of, numerical or computational approach depending on the model at stake. So why do you expect such model to simplify the dynamics and allow you to solve it analytically? It's because, as in any mean field approximation, you hope for asymptotic independence. Okay. So here I just show it numerically. I'm looking at the correlation between neurons in a finite replica model, and I'm just showing that the correlation vanishes very quickly with the number of replica. And this is due to the fact that by randomizing interaction, the likelihood of 2 neurons to talk to one another regularly is scaled like 1 over the number of replica. So they behave effectively almost independently. And actually, we have more than this. We have basically what we call the Poisson hypothesis, which is that in this sort of limit, if you are a neuron of a particular flavor in one replica, and you look at the spike you're receiving from a neuron of another flavor all across the replica, you will see that basically you experience those spike deliveries as if they were delivered from independent Poisson process. And independence here is with respect to the type of neuron you look across replica. And so that tells you that basically there's hope for analytical tractability because the arrival process of spike is well characterized. So now I need to say that at this stage it's just a Poisson hypothesis because it's been conjectured in many models, and we shown that it holds only for a subset of dynamics. And actually, there's going to be a talk later on this week about proving this Poisson hypothesis for discrete dynamics. But okay, it's intuitively true. It should hold, and it's also observed numerically. Okay, so we're gonna assume that. Now, if you assume the Poisson hypothesis, now you can use your replica framework to hope to solve analytically the approximating dynamics. So here I'm just going to show it, and I'm going to show what it involves usually. What do you have to do computationally to solve a model in the replica mean field limit? And then we'll talk about point 1 and 2. So here is the full dynamic of the interacting system. So it has a joint stationary distribution, and it's complicated because of the interaction. It's hard to do anything about it. But we know a few things about it. So in particular, we know that we could look at the probability generating function associated to those counting variables. And because the underlying model is still fairly simple, it's linear, we know that it should follow some functional equation. And if you walk a little bit around the model, you can see that, okay, this functional equation that the generating function shall satisfy is some PD. Okay, and it's a linear PDE because we've chosen some linear stochastic intensity. And so in principle, if you want a full characterization of the model, you'll have to solve this. So it's impossible, and it's impossible because of the presence of non-local boundary terms here. And those boundary terms are the one mediating interaction. So what does replica mean field does for you? Well, it maps this model onto one where the interaction have basically been erased because you have asymptotic independence. So you erase interaction, and so you map your model onto a collection of independent mean field model, where each mean field problem is of the following flavor. Like you're trying now to solve for a probability distribution of a collection of neurons who will output at some rate beta, knowing that they receive 4 times this output beta because everything is exchangeable. Okay, so that's the simplest possible kind of mean field problem you can come with. And if you look in detail what form that problem takes, you'll see that basically you independent, it's like projecting this equation onto independent direction. And the resulting equation you get now is a simple ODE along each direction corresponding to each neuron. So now it's an ODE, there's nothing scary about that, you can solve it. The only potential difficulty about it is that now each ODE is parameterized by that red beta. That is perhaps the simplest possible characteristic of the dynamic we would like to characterize, okay? And so when you do a replica mean field approach, the first step is to get to those equations, which there's a recipe to get them. You're not gonna be able to always get them, that's gonna depend about the model. And then once you have those equations, the goal is you have a scheme, a mean field scheme, and the goal is to solve for it. And you want to solve for it efficiently because you want to be able to do numerics. So I'm going to just show you what it looks like in this simple case where it's an ODE-based scheme. So okay, it's yes. Do you have any question? No. Okay.
No question.
So yes.
No question. Don't worry.
Oh, no question. Okay, sorry. So okay, so it's always going to be the same. So what's going to happen is you're always going to have way too many solutions than you need at first. And so, for instance, in that case, you can look at the generic solution of this simple ODE, and you will see that, okay, there are tons of them, okay? Like you need an initial condition, and there's an infinite amount of possibility for them. But there's also this free parameter dependence on beta. So, but what those solutions have that is of interest for you is that they all have a pole in 0. And so that tells you that, okay, you have too many solutions because you have singular solution, and you should not care about singular solution because you're after a probability generating function which shall be analytic and well behave. So it's always going to be the same. When you do a replica mean field limit, you always have too many solutions, but you can throw almost all of them because you have to impose a requirement of analyticity. So in this case, there's only one form for solution that are no pole in 0. So, and that's that form. And it still depends parametrically on beta, right? And you're after the value of beta. So then all you have to do is to impose the normalization condition on that any generating function should satisfy. And then now you have a simple equation on beta, and that's it. you've defined the spiking rate that your neuron assume in its stationary dynamic. And from this knowledge of the spiking rate, you can actually guess all the moments of the dynamics. Okay, so that characterizes entirely the full dynamics. And so that's it. It's always going to be the same. What's going to change is that you're going to have more complicated equation to start with, and you're going to have to be a bit more creative to throw away the non-analytical solution. Okay, but one of the great benefit of this kind of approach is that you can apply that independent of the structure of the network. And so here I'm just showing an example about what you can do for those kind of model. So here I'm considering a feed-forward network. Okay. And I'm just comparing to exact simulation the rate that you obtain by using the replica mean field trick, and I compare it to also the classical mean field trick. And in that case, there are dense connection from one layer to another. So basically, there's both of them work very well, and there's no real benefit to go to a replica mean field rate, and that is because connections are dense, so that there's a lot of averaging going out, and it doesn't really matter that you keep track of the finite structure of the network. So it doesn't really matter that you know that you only have like, I don't know, 5 neurons that you receive input from on average. But now if you go to a feed-forward model where like now it's sparse and your activity is dominated by a few sets of large connection, well, then you see that's when like the replica mean field trick becomes interesting, because now you outperform the classical mean field limit. And so this is just to like say that, okay, we don't claim that replica mean field is always good. It's only going to work for a subset of network, and it's only going to matter if you in a regime where actually finite size effect will be dominant. Okay, so that's it for the for basically the approach and like what is at stake when you're trying to solve it. So now let's see if you can do anything with this kind of approach to tackle point 1 and point 2. Point 1 being correlation in the network, and point 2 being like how to deal with metastable dynamics. Okay. So if you want to try to get a go at the correlation structure, you're going to be led to a, yes, no, you're going to be led to something we call pair replica models. Do you have any question at this stage?
No, there is no question.
Am I going too quickly?
There is no question, I think.
All right. Is there anybody in the room?
Yeah. There is someone. You hear me or? Yeah,
no, I hear you, of course. No, okay. I'm joking.
Good. Don't worry, we are with you. You're not alone.
Okay, good. Okay, so yes. So back to the correlation problem. So replica mean field is a mean field approach, so it erases correlation. So it seems mysterious how you could like try to get at correlation within that framework. However, there might be a way, and that's the way we like currently following. And so it all comes from a simple idea. So the replica mean field limit that I exposed to you so far is based on the fact that the individual constituent in each replica that interact randomly with one another are the neurons, okay? But in principle, you could use the replica or replica trick for a replica model for which the elementary constituents are not individual neurons, but have more structure. And in particular, you could do that for pairs of neurons. So here, what I'm showing is a replica model where I take all the possible pairs that I can form in my original network. And now I'm having those pairs being the elementary constituent interacting with one another. So what does that mean? Is that if that blue guy spikes, it's engaged with that green guy as a pair. So it's always going to send a signal to that green guy. But then when he wants to talk to a red guy, he's going to pick that red guy uniformly and independently across pairs that don't involve blue guys, okay? And so basically it's a different way to split your network. And there's a mean field problem corresponding to this. And now it's a mean field that involves a pair, okay? So you could play the same game as I told you before, and you would end up with basically a self-consistent functional equation that characterizes the generating function of the pair. But now, because you have a pair, it's going to be a PD. Okay, a two-dimensional PD. And you recover like those non-local boundary terms that are kind of annoying. But it so happened that for the pairs, and only for pair model actually, you can still solve it. there are tricks. I'm not getting into it. I know François Baccelli has given a talk about that recently, and it's on YouTube, so you can go for the details. The point is, you can find a parameterization of G in terms of two functions that satisfy a coupled Volterra equation, and these are amenable to numerics. And so what can you get with that? So the first thing you can get with that is that now you can have input-output function that are not tied to a single neuron, but to a pair of neurons. And so that means like you give me the input to the pair. So of course I have to assume that I'm in a limit where having independent Poissonian input is relevant. Well, then I can tell you what's not only what are the output rates of the pair of neurons, I can tell you the correlation, the pairwise correlation between neurons. And so why such a thing may be of interest? Well, it's of interest because you know, a lot of like the plasticity rules that people use are based on a correlation measure between neurons. So that's I believe very interesting to have those input-output function available if you're in the business of designing plasticity rule and exploring what will be the impact over the correlation of the network. So what else? Then of course you have more than input-output function. You can also look at what brings you the ability to predict correlation in the network in terms of predicting the dynamics of the network. And so here I'm just showing an example of like a completely arbitrary but chosen, of course, so that it works, architecture, where basically I looked at an architecture of neurons that are engaged into pairwise interaction in a hierarchical fashion. Okay, so that's typically the kind of architecture where I expect the Poisson hypothesis to be valid. And so, and it also exhibits a degree of correlation because I have this strong pairwise structure. And so here what I'm showing is that basically I compare the replica mean field and the pair replica mean field model and show that the rates are better predicted by the pair replica mean field. And that, okay, this improvement in rate prediction is due to capturing essentially accurately the correlation among pairs of neurons, okay? So there are all different realm of like models you can come about based on those pairwise interaction. That's just one kind of model. But you know, you can again like explore that in more detail by going to François' talk online. Now I want to go to the things I'm kind of working on right now with a student who's going to give a talk later on during the talk, and that's looking at whether or not you can extract temporal information from those stationary mean field limits. And so the goal here is to ultimately being able to have like a mean field characterization from which you can infer things such as transition rate, which seems like impossible, but like let's try to do it. And I'm just warning you, we're not going to succeed yet, but I'm just going to show you in which way we think about this. So we're going to do that in the context of a different set of models, which are exponential models. These are perhaps more biophysically relevant models. These are basically the same as the one the counting model I've introduced originally, except that now it's no longer a counting variable. It's going to be a continuously varying variable x. And that variable will reset, just like as the counting variable, but it also will relax toward a base value. And it will do that in an exponential fashion. And also, we're going to allow this variable to take negative value. It can be positive, negative. So it's no longer going to be a proxy for the rate, because now the rate is going to be obtained as an exponential transformation of this internal variable x, okay? And so you can think basically on this x variable as a proxy for membrane voltage. And so now then the quantity of interest that we're going to look at is the moment generating function of x, okay? So now you have a nonlinear model, so things are going to be a little bit more complicated. And if you carry out the analysis, you realize that this moment generating function still satisfy a functional equation, and this functional equation is going to be more difficult to deal with, at least. Why? It's because now, due to the nonlinearity, you're going to have some non-local term that appears. And actually, this equation is going to become a delay equation, okay? And so this seems a big challenge because delay differential equations are by nature infinite dimensional, and it's very few of them can be solved analytically. However, here in this case, there's a trick, and the trick will be dealt in more detail by my student later on. Here, I'm just giving you the flavor. The trick is that within this model, you can actually rewrite the delay term under that form. And now you see that H, which come into play in the intensity function here, which you can think of as the base rate, the base level of spiking of a neuron, can be interpreted as a perturbation parameter. When H is equal to 0, this becomes just a plain ODE for which you can solve. And so then when you have this non-zero delay term, you can hope to form a perturbative series of the solution and hope that it has the right analytical property that you are after, so that it represents a physical distribution. And the whole technical analysis is to show that it is indeed the case. Anyway, so it is the case. And so now you can look at mixed population of excitatory and inhibitory neuron, and you can look at them in feed-forward model as well as fully recurrent model. And the mystery here is that when you put inhibition, basically the replicamin field limit works all the time. It's not conditioned only to work in feed-forward structure. It works also in recurrent architecture. And we don't have a good understanding why it's the case, but okay, it seems that that it works. So that's a sanity check. But what we're really after is metastable dynamics. So let's look at a model that exhibit metastability. And this is a model like this. That's the simplest kind of metastable behavior you can think of. That's a bistable behavior. So the architecture is fairly simple, right? You have two groups of neurons, each comprising an excitatory and inhibitory population. And basically, there's going to be a cross inhibition in between the two groups. And if the cross inhibition is weak, there's going to be inherent stochasticity in the model, and basically, you're going to be monostable. But if the cross inhibition becomes strong, then one group being active will essentially silence the other group. And because of the symmetry, only one group at a time can be active. And there's like a stochastic switch between group 1 being dominant or group 2 being dominant. So that's it. So that's the bistable behavior. And so basically the kind of thing you would like is give me an architecture of a model, and are you able to give me some interesting feature about the model, such as transition rate between different states? And so that's a very difficult problem, and it's not a model that will be able to be captured by classical mean field model. It's known already. And so the hope is that by a replica mean field approach, you could capture that. So why would you hope this? It's because the transition from one dominant state to the other is essentially driven by the level of noise that is exhibited by the dominant population while it's up, okay? And so the hope is that if you can characterize the variability of the population when it's dominant, you should be able to estimate the escape rate and then the transition rate. And you can do that in a replica mean field limit, because also it's a mean field limit. It has a degree of variability because you have a Poissonian arrival, a Poissonian spike delivery. So there's always a measure, a sense of what is the variability of the population. And that's by contrast to classical mean field, where everything gets average. So everything is deterministic. Okay, so can we have any chance to actually complete this program? And so here I'm just showing the first slide of the talk of my student, which basically argue that yes, there's hope you can do that. So here, what that slide shows is that you vary the interaction strength in the previous model, and you're going to compare the exact model to the simulation. The exact simulated model to the value you get from the replica mean field ansatz. So the value you get from the ansatz are in solid line, and the simulated value are obtained in open circle. And you vary the interaction strength, so you expect to switch from a monostable regime to the bistable regime. And so what happened for the replica mean field limit exactly? Okay, so the original system that, yes? No, okay. So the original system that you simulate is metastable. So that means it's going to alternate between those up and down states. And so basically you can condition to between up and down and measure conditioning to those up and down state. What is the average rate of firing? So that's what you get there. And you can also do the same thing for the internal variable, and you can compute all the moments of the internal variable. And so then, if you think about the replica mean field limit, it's an infinite size system. And so actually, it will not be metastable, but it will be multi-stable. So what will happen is that the equation that you get in the replica mean field limit will admit multiple solutions when the metastable regime arrives. Okay, and so that's what is plotted here. Like suddenly you have like multiple solutions, and those multiple solutions actually closely follow the value that I estimated in the exact simulation. Okay, and so basically this is just to demonstrate that like you can capture basically what's happening in the real system, and also what's happening in like the higher moment of the distribution of the system via the replica mean field. So then we're not there at like being able to estimate or predict some transition rate, but we're trying to get there. But you can also like estimate some other interesting features. So for instance, here I just show an example of something that I find interesting. And it's just saying like, okay, so you have a system that you can explore, and you can exhibit the dependence on the structure of the system. So here that's synaptic weight due to excitation, due to inhibition. I can change them, and I have a phase space. And in red, I have bistability. In blue, I have the monostable regime. And now I can ask a question such as the following: like, okay, let me look at all those values for which I'm going to be just at the transition between bistable and monostable regime, and see how it scales with k, where k is the number, the finite size number of neuron in each of my cluster. Okay, and then, like, if you ask this question, you would realize that, oh, if I bound my dynamic to remain on the front, separating those two regimes, I have to choose k that scales like an inverse square root, just as like the balance model. And so then you can gain, like, you can ask questions that I think would be extremely hard to answer if you were just conducting a brute force analysis by simulating exact models. Like, you really have like a few knobs, and you can basically estimate scaling behavior that would be very hard to characterize otherwise. Okay, I think I'm going to stop here. I just want to remind you that this is work done in very close collaboration with François Baccelli, which is now he actually has left UT last time I heard, and he's now fully at Henri. The two talks I mentioned are going to follow later this week will be given by Michel Davidoff and Louis Anliu, and here are the relevant references and my funding. And I think that's pretty much it. I'll be happy to take your question.
Thank you very much. Is there any questions or comments?
Thank you for this nice talk. I have a question regarding the slide just before that, I think. Yes, exactly.
Wait, this one?
Okay. Metastable dynamics, yes. No, the previous one. Yeah, exactly, that one. So okay. Here, when I'm looking at the connectivity between those two pools, or those two groups, what I was expecting was rather, if we consider, for example, okay, in my interpretation, each group is like a column, like a cortical column. So what I was expecting that, okay, cross group connections would have been rather based on excitatory connections rather than inhibitory connections, or both of them at the same time. So what is the motivation, or is there a motivation or a physiological motivation behind this connectivity pattern, or?
Okay, so I think I get the question. So it's about like, is there any motivation, biological motivation for considering such an architecture, right?
Exactly.
So yeah. So actually, there is one. Believe it or not, but it's not. I wouldn't say it's related to a physiological model. It's related to a psychology study who studies a perception rivalry. Okay. so these are like, you know, like imagine you look at a cube, okay, in 3D, and it's projecting in 2D, and you have to decide whether and you color how do you call that, a vertex of the cube, and either you see the cube popping up to you or you see it popping up far away from you. And so so basically people have been looking at rivalry between like those percepts, and the people have looked at like a neural network that basically exhibit that architecture, and whose dynamics will display the same kind of feature as you would see in this rivalry task. So for instance, they look at the distribution of switching time, and they show that, oh, it seems to like match what has been measured in psychophysics and things like that. So it's more like a phenomenological model. It's not really physiological model.
Okay, okay, I see. Thanks a lot. And one last thing. Did you consider, or let me ask in this way, is there a difference in the weightings of connections which we see in the slides? I mean, between, for example...
Oh, yeah. Like these are... so, yeah. So, these are not the same weights at all. Like, you can change them. So, if you look at the phase space, I have mu e and mu I. So, like, there's a mirror symmetry between the two sets of neurons, right? But this is mu e. That is mu I, and they change, right? I explore the space. Then you can put some variability on it. You can put some heterogeneity. Sorry, like there's it will work just fine.
Okay. Okay. Thanks a lot.
Okay.
Okay. There is a question on internet. So Bruno Cessac asks, how does your approach compare to the Fojras-Maclaurin results on mean-field recurrent models with correlated synaptic weights?
Oh my God, that is way too precise of a question for me to answer. So maybe like, can Bruno tell me what exactly is the approach based on?
Okay, Bruno, could you give more explanation, or could you log to the Zoom link so that you ask directly your question?
So if there's no answer, I can tell you what is a claim of originality, if any. Okay, so that's really this idea of like keeping the finite size structure in replicas and multiplying the system. So I don't know like what the paper is referencing to.
This
but basically all those ideas. Sorry, yeah.
Yeah. I think the paper is about another concept of. It's not really a mean field in the sense that in the limit, the synaptic weights do not vanish, so that you have still correlation in the limit, but you can prove that there is a limit distribution of this kind of. I think.
Okay. I would have to look. I would have to look. So yeah, I guess I would have.
So okay.
Here I am. Can you hear me?
Yes.
Bruno is arriving. Perfect.
Sorry, but it's just impossible to ask questions from YouTube, and we are asked not to connect on Zoom, so it's a bit complicated. Anyway, if you can hear me, that's fine. Hello, Thibault. Nice to meet you. Yes. Well, in the beginning of your talk, you have mentioned a couple of work on mean-field method. Actually, you have forgotten a couple of them.
Yeah, I'm sure. I'm not. I don't claim.
But it's a bit pity because physicists have done so many things. But anyway, here I am talking about the work of mathematicians, actually Olivier Fojras and Jean McLaurin. Maybe Olivier is there and he could talk instead of me. But I mean, they were concerned by the point that you mentioned in the beginning, the fact that in general, the synaptic weight in the approach have no correlation, so you get some propagations of chaos and some nice things which make the mean-field work. But they consider seriously the problem when you have a correlated synaptic weight well, we need, of course, that correlation of respect to a certain number of conditions in order to get a Gaussian limit. But using large deviations theory, they were able to obtain in the midfield limit a nice equation for correlated populations. And I was asking myself if, because you have also equation a bit complex in terms of which involve also correlation, I was asking myself if there are any link between these two approaches.
Yeah, so you said like it's based on having correlated synaptic weights?
Yes.
So you mean, but the synaptic weights are like changing, so like they're evolving?
No, sorry, it's a quenched disorder. So
I see.
Yes, they are fixed, but correlated.
Okay, so there's averaging going on?
Yeah. Well,
okay,
actually, there is there is a propagation of chaos. So I think Olivier will confirm, but I think that there is an almost sure convergence result, which means that well, you are a bit above averaging.
Yes, so there's no averaging here, right? Like here it's like at fixed structure, I get a set of equations that can be solved. So like I would say like if there's a benefit to the kind of approach we propose is that we don't ever need like those results, those equations we get never requires averaging. It's just stationary limit.
I'm sure of that. My question is, do the equations that you obtain compare in some sense? Are there any link? Because I mean, there are not so many people studying correlated problems, so it would be interesting to make the link and at least to mention them. Yeah.
Okay. I'll be more than happy to mention them.
Yeah. Thank you.
All right.
I had a small question. For instance, where you show that the firing rate is better explained by RMF than just MF on the plots, yeah. Do you have any estimate on what is the error that you are making with respect to the true system? What does contribute to the error? Do you have an idea of the discrepancy?
So like obviously here I'm like, I mean, obviously it's not obvious, but like I hope I mentioned it when I was talking. I'm cheating in the sense that I confine myself to feed-forward dynamics, right, or feed-forward structure. So basically, when you are in the case where the replica mean field limit is supposed to outperform the classical mean field limit, is that sparse case at the bottom, right? And so whenever you miss something, it's because there's still residual correlation, and that's because well, there's that, okay, there's residual correlation, but there's also another thing, is that in this replica mean field limit, you assume that the input is Poisson, right? So if you receive the input from a previous layer, that would mean that the previous layer is spiking in a Poisson fashion. It's not a Poisson process, because, right, it has a non-constant stochastic intensity. So something that you will always carry around is, even if you are perfect independence, is that error from assuming that the input is Poissonian when it's not, even so it's uncorrelated. Does that make sense?
Okay, but could you quantify it in the sense that, I don't know, if mu is very small, it tends to be, I don't know if it's mu or beta, I'm lost right now, but do you see, does it mean that the smallest interaction, the better the estimation, or you see, do you know exactly what is
I don't have,
how to quantify it?
I don't have it in a satisfying manner, right? I could give you my intuition maybe, but yeah, no, I haven't quantified systematically the error.
Okay, because in a sense it seems like it's like a limited development of the true system, right? Like layer after layer, you can incorporate more higher and higher moments somehow between your neurons. And that, if you control like the rest of the series, you know a bit what you're losing.
Yes, you can come with a hierarchy of equation depending on you can decompose your network into like individual neurons, doublet of neurons, triplet, quadruplet, and then you will have a whole hierarchy of equation. But this is something we'd like to characterize. We haven't done it yet.
Okay. Thank you.
But yeah, there's a whole we're going step by step.
Okay. Thank you. I think, one last question, and after that we need to move.
So, if I understood correctly, you talk about two models. So there was the first GL models, and then there was another version with this spontaneous relaxation toward some basal level of the membrane potentials, which in biological terms relate to electrical synapses. And then you talk about metastability. So if I understood correctly, metastability, it was in this second model, right?
Yes, yes, and it's at the network level. Yeah.
And so, do you think this electrical synapses effect is necessary to observe this metastable behavior or not?
No. So the metastable behavior, the real crucial point is just to have inhibitory neurons, basically. And if you do I mean, in my experience, okay, and if you do a spiking model with an intensity function, you need a nonlinearity because this is just the way. And basically, once you have a nonlinearity, you can come up fairly easily with metastable behavior. I mean, that's what. I mean, when I did the modeling of the exact model, it's fairly easy to have an exact model that would exhibit metastability, as long as you allow inhibitory population.
Okay, thank you.
Does that make sense? And if you have inhibitory population, you need a nonlinear rate function.
Okay, thank you.
Okay.
Thank you very much, Thibaut.
All right, thank you. Okay, I'm going to stop sharing. All right.
And now we pass to the last speaker. I don't know if you... and couldn't make it and even be there online. So he's really sorry for that. He wanted me to show you his web. So he's fine. Don't worry, he's fine. He's just having medical examination right now. But he was able to make a record of the talk just before and after the ski accident. So I think he's all right. But still, because of that, he wanted me to tell you that there is a lot of stuff on his webpage, and if you are interested in anything he's saying in his talk, and there is a lot, because he asked me to watch the video before, and it's a very nice talk. You should stay, even if he's not there, and there are a lot of information. You can go on his webpage to have more information, and you can also ask him by emails, of course, etc., etc. So let me just play the movie, and after that, if you have questions, well, maybe some of the questions we can try collectively to answer or think about it. But if they are too specific, maybe you should ask it to Rob by email, okay?
Firing rates.
Wait a minute.
The result recent improvements in multi-electrode recording technologies have led many people to work on data analytic methods for identifying neurophysiological interactions among distinct parts of the brain. Recent improvements in multi-electrode recording technologies have led many people to work on data analytic methods for identifying neurophysiological interactions among distinct parts of the brain. Our research is motivated by what seems to be a somewhat unusual perspective derived from my lengthy experience as a statistician. My primary research support for more than 20 years has been from the National Institute of Mental Health.
Yes, could you put the sound louder? Sorry for that, we don't hear the sound in the room. We don't know how it goes by Zoom or by YouTube. So could you just tell me if you were hearing anything? Well, on Zoom I'm alone, so I don't know. Maybe everybody left. Ouais, okay.
And I've most recently pitched this by suggesting new statistical methods could lead to improved treatment strategies involving interventions that are precise in space and time. It seems to me that 50 years from now, treatments for mental health disorders will have advanced far beyond where they are today. But to get there, neuroscience will have to go beyond simple static circuit diagrams that depict interactions among brain areas without describing the flow of information while the organism accomplishes some behavior. This kind of static diagram displays the different brain areas that may be interacting when an organism carries out a task, in the case of this diagram, some rewarded behavior. But the behavior is constantly evolving and brain activity is constantly changing. The diagram doesn't show us at what point in time particular pathways are involved. It gives us no notion of sequencing across time. That is, to make dramatic improvements, the field must not only say what circuit should be disrupted by an intervention, but also more precisely where and when, relative to behavior, disruption should take place. Improved characterizations would use timing relationships, consider both local and cross-area computation, and describe cross-population dynamics. The first of three projects I want to summarize for you very briefly is concerned with spike trains, hundreds of them recorded simultaneously across 6 arrays in about 10 brain areas involved in visual processing in response to visual stimuli, specifically drifting gratings of varying orientations and frequencies. In this initial article, we focused on 3 brain areas, V1, LM, and AL. V1, primary visual cortex, feeds into the higher visual areas, LM and AL. Here are smoothed histograms displaying neural activity aggregated across trials and across the dozens of neurons recorded from each area. Each curve displays a population firing rate function as it evolves over time. The most prominent features of these curves are 2 peaks in firing rate labeled 1 and 2, with the first occurring much earlier than the second. One would think that these evoked responses in neural populations could tell us a lot about the interactions among areas. In the trial averaged firing rate curves, peak 1 seems to occur earlier for area V1 than for the other 2 areas, as we would anticipate. And we would also expect that this evoked response would occur earlier in V1 than in the other 2 areas consistently on a trial-by-trial basis. The same might also be true for peak 2, but the situation then is more complicated. The second peak occurs sufficiently late following the stimulus that it likely receives important input from higher cortical areas. So, can we see a relationship between V1 and LM peak 2 timing on a trial-by-trial basis? To answer this question, we will find the time at which each population reaches its maximal firing rate and compare across areas. To do this, you might think you could simply fit curves to the peak on each trial and thereby compute the peak times, but statistically, that doesn't work very well. The panel on the left, labeled B, shows the resulting times at which peak 2 reaches its maximum in V1 and LM, with each black dot representing the pair of times on a given trial. No relationship at all is visible. On the other hand, a Bayesian approach based on a hierarchical model reveals, as shown in the panel on the right, labeled C, a very strong relationship with the posterior distribution of the correlation of peak times portrayed in the inset as a representation of statistical knowledge and uncertainty. Also, the dots in panel C are pretty heavily clustered below the diagonal, and according to the posterior distribution, the peak in LM occurs later than the peak in V1 by about 10 milliseconds. A descriptive name for this kind of analysis is evoked spike response coupling, and it emphasizes its similarity to analysis of evoked response potentials, ERPs. In fact, the methodology assesses coupling based on curves derived from selected neural populations that are specific to experimental condition. Notice the much higher firing rates. The resulting population firing rate curves are sharper, especially for the second peak, which typically attains a maximum firing rate similar to that during the first peak. Also, the most common method of summarizing population responses, Gaussian process factor analysis, GPFA, does find peaks on one of its factors, but it doesn't find coupling. It produces a figure much like panel B. The hierarchical model, which we call an interacting population firing rate model, and the associated computational method, was developed by Yu Chen, a student who recently defended his thesis in our machine learning and neural computation PhD program. It provides denoising and covariation of peak timing, as I've shown you, and it includes clustering of neurons and uses time warping for trial-to-trial variation. Schematically, according to the model, spike trains are generated by point processes with intensity functions that depend on one of three templates, that is, their template functions, according to neuron membership indicators. Only one template function, the interacting population template, is involved in cross-area communication. The other two are there to absorb the majority of neurons that do not participate. The non-parametric template fitting, along with the clustering of neurons, is carried out for each brain area and each experimental condition. A small number of features, including the times of the two peaks, capture trial-to-trial variation of the kind of covariation I just showed you. Importantly, the method is multivariate, so we can look at graphical structure based on partial correlation. For example, we know that V1 feeds into LM and AL, but these areas also get input directly from the thalamus and from other visual areas, which could be especially relevant to the timing of peak 2. As I said, by the time of peak 2, feedback loops are likely to be operating. So what happens when we examine the correlation of timing in two areas, such as LM and AL, after conditioning on timing in the third, in that case V1? Before conditioning on timing in the third area, panel A shows very high correlation of the peak timing of LM and AL. Panel B shows a little smaller but still very high correlation of the peak 2 timing in V1 and AL. Panel C shows that after conditioning on timing in V1, the correlation of timing in LM and AL is somewhat reduced compared to what's in panel A, but it's still very large. In contrast, panel D shows that after conditioning on timing in LM, the correlation of timing in V1 and AL is dramatically reduced compared to panel B. There's no longer any visible relationship between them. These are interesting relationships, which we have replicated in a different mouse. And of course, plots are all easily obtained from the posterior distribution of the covariance matrix for the trial varying features. Let me now turn to the second project, torus graphs for multivariate phase coupling, where we analyze local field potentials that are oscillating. As you know, oscillating local field potentials have been observed under a variety of circumstances, and there are plausible mechanistic network models to explain them. One leading theory is that they facilitate communication across brain areas by forcing populations of neurons to spike at nearly the same time. Because neuron membrane voltage decays fairly rapidly, such nearly synchronous spiking could increase the firing rate of a downstream population. Earl Miller's lab at MIT has been especially interested in oscillations during learning and memory tasks, with several papers focusing on the interaction between prefrontal cortex and the hippocampus, which are widely considered to be crucially involved. We analyzed some of Brinkat and Miller's LFP data, bandpass filtering it as they had done at 16 Hz. We then selected a particular single point in time on each trial at which to measure the phase from each electrode, and looked for phase coupling, meaning a fairly consistent phase difference across repeated trials which would indicate coordinated activity across the brain areas. The standard measure of phase coupling is called phase locking value. It's intuitive, and as we show in the supplementary material of our paper, under special circumstances it does estimate a natural analog of correlation for angles, which in statistics we call circular data because an angle specifies a point on a circle. However, phase locking value is a pairwise measurement. While Brinkat and Miller had 24 electrodes across PFC in 3 regions of the hippocampus, CA3, DJ, dentate gyrus, and subiculum, so we wanted a multivariate method, which would give us a more complete picture of interdependence as provided by a graphical model. That is, we wanted a placement of edges connecting the 24 nodes shown here on the right, where the edges indicate statistical dependence. We can construct a set of nodes and edges using phase locking value, but that doesn't give us a probabilistic graphical model, meaning that the absence of an edge using phase locking value does not correspond to conditional independence given all the other nodes. We wanted instead a general circular analog to partial correlation. In some circumstances, a Gaussian graphical model based on the multivariate normal distribution might be satisfactory, but not here. Although these experiments have a well-defined start time on each trial, that doesn't do much to set the phase, so the phase angles across trials are fairly uniformly distributed. To build a graphical model, we have to respect the topological distinction between a circle and the real line. Here's some data simulated to look similar to real data. Real data are shown in the small colored picture to the right, where 2 phase angles were drawn repeatedly, with each being roughly uniformly distributed but with a pretty consistent phase difference. x1 is leading x2. Note the seemingly weird data points in the upper left corner of the plots. When x1 goes a small amount, c, past pi, 180 degrees, pi plus c is replaced by minus pi plus c. Meanwhile, x2, lagging behind x1, hasn't yet gotten to pi. So it's easy to explain what's going on up in the left-hand corner. But the weirdness is an indication that standard xy coordinates are not really appropriate. It's more natural mathematically to consider the product of 2 circles, which is a torus, with one angle identifying rotation in the direction shown here of the blue arrow, and the other angle identifying rotation in the red direction. After smoothing the simulated data to get the color density representations shown here, we see that the weirdness in the plane is gone when it appears on the torus. One of the students made this animation to show more clearly what's going on. The red dot indicates corresponding points in the 2 representations. The torus is clearly a more elegant representation, and to get a 24-dimensional analog for phase coupling analysis, it is statistically natural to develop an exponential family on the multidimensional torus, which we call a torus graph. More specifically, we consider the full exponential family on the multidimensional torus having all 2-way interactions. A torus graph is a subfamily having some specified set of 2-way interactions, which can be represented as edges between the nodes of a graph. The 2 PhD students, Natalie Klein and Josue Oriana, did a very thorough job of establishing the properties and methodology, so there are many interesting aspects of the work. But the key point here is that the absence of an edge in torus graphs corresponds to conditional independence given all the other nodes. In other words, torus graphs are probabilistic graphical models. Again, a graph based on PLV, the pairwise measure, does not have this property. The portion of the linear probe used by Brincat and Miller that recorded from area CA3 had 5 contacts. The torus graph we obtained recapitulates the linear arrangement, with each electrode exhibiting Markovian conditional independence from non-neighboring electrodes conditionally on its neighbors. The PLV graph makes it look like every electrode is dependent on every other electrode. When we consider all 24 electrodes together, again, the torus graph gives us something that's consistent with known anatomy. Prefrontal cortex depends on the dentate gyrus only through its dependence on CA3 and the subiculum. Finally, the last project again considered cross-population coupling, but this time based on local populations that generate the local field potentials, that is, the current sources. According to basic electrostatics, the current source density, or CSD, is the negative Laplacian of the LFP, and the first methods of estimating CSD simply applied numerical second derivatives to the LFP. More recently, it was recognized that CSD estimation is an inverse problem that can be solved using the forward model that maps the CSD to the LFP. This version of the standard forward model comes from our paper where we used A to denote the forward model linear operator. Our statistical perspective led us to model the LFP as the sum of the forward model applied to the CSD plus noise. Based on our experience with LFPs, we then took the CSD to be the sum of 3 components, a non-stationary transient together with a 2-component Gaussian process. Standard forms of Gaussian processes describe fluctuations using a single time constant parameter. Here, we forced the 2 components, the 2 Gaussian process components, to have very different time constants, making one of them relatively slowly varying and the other more rapidly varying. The first was designed to capture trends, and the second, the oscillatory fluctuations we were interested in. So this is a Gaussian process current source density model, and it can reproduce LFP spectral densities extremely well. The paper provides reasons to believe this approach is more accurate and more general than other existing ways to get CSD. And in auditory cortical data, we found 10 Hertz phase coupling across cortical layers in 2 separated columns of cortex that were not apparent in the raw LFP. So those are my 3 examples, and as I've reflected on what binds them together, I've observed that in each of these 3 cases, interactions across neural populations seem to become apparent when interaction and population are defined appropriately. The interacting population firing rate model defined interaction in terms of peak timing and used clustering to select relatively homogeneous populations of neurons, thereby revealing relationships that could not be seen using naive methods. Torus graphs define multivariate phase coupling as a form of interaction, producing relationships unavailable from the pairwise measure. And in the case of Gaussian process current source density estimation, the relevant populations were those we found to be generating the currents. One of the things that makes LFPs hard to work with is that they reflect a superposition of differing voltage patterns from many distinct current sources. Our GPCSD estimation amounts to deconvolving the LFP into spatially distinct components. Also, when I mentioned we found phase coupling across cortical layers, shown here as superficial, medium, and deep on 2 different probes labeled lateral and medial for their locations in primary auditory cortex, that, of course, was again using torus graphs to define interaction. Importantly, we were unable to see this kind of pattern when we examined the raw LFPs. In all these cases, considerable experience and wisdom within the field of statistics informed the design of these approaches. The examples also illustrate the 3 scientific goals I started with: using timing relationships, considering both local and cross-area computation, and describing cross-population dynamics. And when I said we should consider both local and cross-area computation, I had in mind the substantial structure within areas reflected in particular by cortical layering, shown here, that does seem to carry out local computation. The three examples did, in a certain sense, illustrate these three aspects of neural computation. The interacting population firing rate model used timing relationships. Torus graphs describe cross-population dynamics in the form of oscillating circuits. And Gaussian process current source density estimation considered both local and cross-area computation. Local interactions occurred within one of the two probes and are not shown in this figure. But in terms of what I meant the field of neuroscience will have to accomplish, what we did in these papers amounts to taking baby steps along that path. One of the most frustrating issues is the need for good statistical descriptions of cross-population dynamics that can be used in conjunction with mechanistic descriptions. Put differently, we should ask, where are the synergies of statistical and mechanistic mathematical modeling? This was the point of a set of discussions, including 2 international meetings, that led to the review paper cited here. I like the paper a lot, but I found the effort nonetheless disappointing. Not only were there few good examples of the synergy I've been looking for, there were few concrete ideas for bringing together these two sub-disciplines of computational neuroscience. One simple way to start, which I hope to pursue with a couple of students, returns to the commonality among examples I just pointed to and poses an obvious question. Under what circumstances, what assumptions, can that observation be made rigorous and be verified theoretically? We could, I presume, specify assumptions purely statistically. In the case of the interacting firing rate model, we could assume that we recorded samples from homogeneous local populations driven by firing rate functions. We defined interaction using peak timing, which led to well-defined directional timing relationships across areas. But while peak times are intuitive, using them is really a statistical convenience. And we might want to think more generally about forms of dependence between time-varying functions that are driving separate neural populations. There's all kinds of dependence we could dream up statistically between random functions, but they ought to be possible in neural systems. So it would be much more interesting if we could connect to that large body of work that gives mechanistic descriptions of neural population dynamics. But then, our statistical models would not simply be describing variation in data, they would also become part of a mechanistic explanation. And within the field of statistics, this sounds like a fundamental shift in the way we think about the nature of statistical modeling. The nature of statistical modeling is something that statisticians have discussed for many years, and I've added my own comments in several places. I would like to draw your attention especially to a recent contribution which acknowledges the commonalities of statistics and machine learning, including artificial intelligence, but then tries to specify the continuing value of this perspective advanced by the field of statistics. Common wisdom, correctly in my view, places statistical models at the center of modern statistics. However, with all the thought I've put into this across many years, my appreciation for the value of mechanistic thinking in statistical modeling is not something I've articulated. Concerning the possibility that models provide explanatory mechanisms, statisticians remain agnostic and skeptical, whereas neuroscientists are boldly adventurous. I've come to realize we need both attitudes. And what's shaped my thinking, as much as anything, has been my experience over the past 10 years teaching a basic graduate course in computational neuroscience. I call the course Statistical Models of the Brain with the hope of emphasizing that it is not a course in data analysis, but rather a course that surveys many leading roles played by statistical models in explanations of how the brain works. By the way, if you go to that website, you will find that there is a video for nearly every lecture, and in fact the videos are very short, they're concise, so you may want to have a look. Witnessing the way neuroscientists use statistical models for explanations makes me think there may well be some benefit for statisticians to look more favorably on the notion that models can, in some sense, specify mechanisms, at least in certain circumstances. But what circumstances? Perhaps I'll figure out something coherent to say about this and write about it. We'll see. In any case, I do hope some of you might consider this a topic worth pondering. Thank you, and sorry I couldn't be there.
Okay, so I don't know, do you have bold questions or bold models to propose? Maybe also we could keep a discussion about what is said for tomorrow's discussion. I don't know, as you want. But if you have comments that you want to share right now, it's the moment. No? Just looking on the... So, okay, let's say that we keep this into mind for the night and we discuss all of that tomorrow. So tomorrow morning we will have 3 talks by young people who are doing stochastic stuff around neurons. And then we will have this discussion that is led by Ingrid and Alex. And basically every morning we will have a discussion with the main speaker of the last afternoon, of the previous afternoon, so that we can discuss the importance of the tools, and we can also discuss with biologists about here what statistics means for neuroscientists maybe and how they can help, and maybe discuss about what we heard Robert said, because I think it was a good thing to think of about the boldness of neuroscientists and the agnosticness of the skepticism of statisticians. Maybe there is something in between that is nice. Okay, so we meet everybody tomorrow at 10, and thank you all for being here today. Goodbye.